International Conference on Molecular Insights into Cancer and Autoimmune Disorder. On behalf of the organizing committee at Ramakrishna Mission Belur Vidya Mandir, I welcome all of you to this Vivekananda Auditorium. We'll start our program with Vedic chanting performed by the students of Department of Microbiology and Zoology.
Om Shri Gurubhyo Namaha Harihi Om Sahara Bhavatu Saharo Bhunaktu Saha Viryam Karvavahai Tejasvinavadhitamastu ma Vidvishavai. Om Shanti Shanti Shanti. Om Shamno Mitra Shamvarunah Shamno Bhavaktaryama Shamno Indro Brihaspatihi Shamno Vishnu Rurukramah Namo Brahmane Namaste Vayo Tvameva Pratyaksham Brahmasi Tvameva Pratyaksham Brahma Vadishyami Ritam Vadishyami Satyam Vadishyami Tanmamavatu Tadvaktaramavatu Avatu Mam Avatu Vaktaram. Om Shanti Shanti Shanti. Om Bhadram Karnevi Shriniyamadevah Bhadram Pashyemakshaviryatraha Sthirairangaye Stuštuvagam Sastanuvihi Vaishema Devahitayadayuhu Swasti no Indro Vriddhashravah Swasti na Pusha Vishwavedaha Swasti nastaksho Arishtanemihi Swasti no Brihaspatir Dadhatu. Om Shanti Shanti Shanti. Om Purnamada Purnamidam Purnat Purnamudachyate Purnasya Purnamadaya Purnamevavashishyate. Om Shanti Shanti Shanti. Harihi Om Tat Sat. Shri Ramakrishna Arpanamastu.
Thank you for the serene rendition. Moving next, we'll now have the inaugural address by our respected Principal Maharaj Swami Mahapragyananda, and I request Maharaj to kindly deliver his address.
Om Namo Bhagavate Shri Ramakrishnaya. Very good morning, everybody. Today we all assembled here to attend this one-day international conference on molecular insights into cancer and autoimmune disorder. On behalf of Vidya Mandira Parivar, I'm very happy to see you all, especially the students, faculty members, distinguished guests. I welcome everybody. On the dais, present Dr. Triparna Singh. She has come all along from United States to join us today. Dr. Singh is an Associate Professor, Department of Oncological Science, is a Lung Cancer Division, Asian School of Medicine at Mount Sinai, New York. We have another resource person who is also present in dais, Dr. Orgho Chattopadhyay. He is right now the Academic Director, Asian Institute of Immunology and Rheumatology, and Our third speaker, resource person, Dr. Uday Bandyopadhyay. He is the ex-director, Bose Institute, Kolkata. I cordially welcome all our three speakers today. Welcome, sir and madam, to Vidya Mandira, the Temple of Learning. These two sessions will be, this three presentation has been divided into two sessions, two academic or scientific sessions. The first session will be chaired by Dr. Subir Chandra Dasgupta, who is one of the faculty member is the ex-professor, Department of Zoology, Maulana Azad College, and right now Professor Dasgupta is a Professor Emeritus Honorary in Department of Zoology, Vidya Mandira. And the second session will be chaired by Dr. Pratap Kumar Das, a renowned scientist, retired, of course, in an Institute of Chemical Biology, and Dr. Das is one of the illustrious alumni of this institution. I cordially welcome both of them, sirs, and especially today faculty members from different colleges, universities are present here. I cordially welcome, I extend a very warm welcome to all of them. And the participant, students from different colleges and universities who have come here to attend this seminar, this conference, and also I'm very happy that they have brought their presentations. It is very nice to see. We always encourage we all, faculty members, we feel very happy to see when across the colleges, the students, especially of undergraduate and postgraduate level, they are doing those small projects, So this poster presentation is a very unique thing, and number of posters have already been placed outside. So I congratulate and I cordially welcome to these participants, students and poster presenters. Finally, I congratulate especially the faculty members of both the departments, microbiology and zoology, who worked actually tirelessly since last 1 month for entire planning, and especially the students also. These two are undergraduate departments in Vidya Mandira, and our students, undergraduate students also, they supported, they are definitely the part and parcel of this, they were and they are the part and parcel of this entire planning. So congratulate the faculty members, head of the departments, especially microbiology and zoology, and our students. I will wrap up with just a couple of words. Those who are coming for the first time this place, Vidya Mandira, for them, I would like to intimate you one fact. This very name, Vidya Mandira, the place where we are sitting right now, this is actually the dream of Swami Vivekananda. This very word, Vidya Mandira, was his coinage. In his physical lifetime, Swami Vivekananda envisioned one academic institution.
Scientific leaders across India and abroad to delve into cutting-edge research, share insights, and foster meaningful discussions on the molecular mechanisms underlying cancer and autoimmune disorder. We are indeed honored to host over 350 participants from across about 50 different academic institutions representing a vibrant of public and private universities, postgraduate and undergraduate colleges. The overwhelming response reaffirms the urgency and importance of collaborative academic efforts in addressing some of the most complex health challenges of our time. We are especially privileged to welcome our keynote speaker, Dr. Triparna Sen, and other distinguished luminaries, Dr. Orgho Chattopadhyay, Professor Dr. Uday Bandyopadhyay, Professor Dr. Shubir Chandra Dasgupta, and Dr. Pratap Kumar Das, whose work continues to inspire and shape the future of biomedical science and healthcare. Rooted in the ideals of Swami Vivekananda, as Maharaj said, Ramakrishna Mission Vidya Mandira believes in a holistic education that nurtures the head, heart, and hand. This conference is a humble step in that direction, an effort to harmonize scientific inquiry with a greater cause of social welfare. We look forward to an intellectually stimulating and spiritually uplifting day ahead. And once again, I, Abhishek Banerjee, on behalf of this organizing committee, extend a very warm welcome to each of you, and may this day filled with curiosity and collaboration. Thank you. So now we will take this opportunity to felicitate our speakers, and I would request our Maharaj to kindly do the honor. So first, we are going to felicitate our keynote speaker, Dr. Triparna Sen. Dr. Sen is an associate professor in the Department of Oncological Science, the Tisch Cancer Institute, and co-director of Lungs Cancer PDX program at Icahn School of Medicine at Mount Sinai, USA. We next felicitate our invited speaker, Dr. Orgho Chattopadhyay. Dr. Orgho Chattopadhyay is a doctor at and director at Asian Institute of Immunology and Hematology. We next have our speaker, Professor Dr. Uday Bandyopadhyay, who is the ex-director of Bose Institute, Kolkata. Thank you, Maharaj. We formally come to the end of the inaugural session. We'll now start our scientific session 1. Just give us a few minutes to prepare the stage. Thank you. It's my honor to introduce Dr. Dasgupta, who is an eminent academician and accomplished researcher and one of the most respected names in the field of zoology, pharmacology, and toxicology. Professor Dasgupta has had a distinguished teaching and research career spanning nearly 4 decades, including over 21 years of postgraduate teaching and over 32 years of undergraduate level teaching. He retired as Professor of Zoology from Maulana Azad College, Kolkata, and currently serves as an Honorary Emeritus Professor at the Department of Zoology here at Ramakrishna Mission Vidya Mandir. His research interests lies in the cutting-edge toxicology, pharmacology, and screening of bioactive compounds, and over the years he has mentored 13 PhD students. He has over 17 major research projects funded by prestigious agencies like ICMR, CSIR, DST, DRDO, and name more than that. And he also has 2 patents, one which is an international US patent. He has over 90 peer-reviewed publications, and beyond research, he has served in numerous academic and advisory capacities, including curriculum and development, editorial boards, biosafety ethic panels, as an advisor to DBT Star College scheme at national level. He is also a life fellow of the Zoological Society and several other scientific bodies. We truly believe to have him, we are truly privileged to have him with us today as a chairperson. Please join me in warmly welcoming Dr. Shubir Chandra Dasgupta. Sir.
Our keynote speaker, Dr. Triparna Sen. Please, madam. It is with a great honor that we welcome Dr. Triparna Sen as our keynote speaker. Dr. Sen is an associate professor in the Department of Oncological Sciences at the Icahn School of Medicine at Mount Sinai, Newark, and serves as a co-director of the Lung Cancer PDX program. With a PhD in cancer biology from the Jadavpur University and a master's in genetics from the University of Calcutta, her academic journey underscores her deep-rooted commitment to cancer research. Dr. Sen is focused on understanding the biology and the development of new treatment paradigm for classical, clinically relevant molecular subject of lung cancer like non-small cell lung cancer and small cell lung cancer. Dr. Sen is a pioneer in the field of lung cancer research and has driven fundamental advances in the understanding and characterization of SCLC, the most aggressive form of the lung cancer. Previously, Dr. Sen identified that DNA damage response aggregates proteins are effective therapeutic target for small cell lung cancer treatment, and that targeting DDR promotes anti-tumor immunity through the activation of innate immune signaling. Dr. Sen received several prestigious awards including AAAS Martin and Rose Watlett Cancer Research Award 2021, 40 under 40 in Cancer Emerging Leader Award in 2020, AACR Women in Cancer Research Scholar, AACR Scholar in Training Award, Jeffrey Lee Cousin Fellowship, and Immuno-Oncology Young Investigator Award. Dr. Sen is invited to deliver talks and educational session at major national and international meetings. Dr. Sen has received independent grant funding from NIH, and currently she has 3 prestigious ROI grants to run her lab at Mount Sinai, a rare and distinguished honor. She has also received the grants from the Department of Defense, Lung Cancer Research Program, Lung Cancer Research Foundation, and the International Association for the Study of the Lung Cancer. She is the author of several peer-reviewed scientific manuscripts, and she is an active member of IASLC and the American Association of Cancer Research Center. Her scholarly excellence is evident from his over 50 peer-reviewed publications in high-impact factor journals like Cancer Discovery, Lancet Oncology, Nature Communication, Cell Report, Medicine, Science Advances, Cell Report, Clinical Cancer Research, to name a few, and a Google Scholar H-index of 40 with over 6,700 citations. Due to her expertise and contributions, she has been nominated to be the conference co-chair of the hot topic small cell lung cancer meeting supported by the International Association for the Study of the Lung Cancer in October 2021. Dr. Sen's dedication to translating complex scientific discovery into clinical application exemplifies her commitment to improving patients' outcome. We are privileged to have her share her insight and expertise with us today. Please join me in giving a very warm welcome to Dr. Triporna Sen. Please.
Hi. I hope I'm audible. Good morning, everyone. It's an absolute pleasure to be here. And Dr. Dasgupta, that was an incredibly generous introduction. Thank you so much. First of all, I would like to thank the organizing committee, Dr. Shami Mahapragyanando, the entire staff, and Dr. Abhishek Banerjee, who happens to be, and I'm very proud and privileged to call him my mentee. I'm very, very honored to be here today, and thank you so much for giving me this honor to deliver the keynote address. So as Dr. Dasgupta mentioned, I'm a translational researcher, so I run a lab, and our real goal is to identify novel biology and therapies for one of the most aggressive forms of cancer, lung cancer. So for those of you who don't know, lung cancer is the leading cause of cancer-related mortality, not just in the United States, but also globally. Most people who are affected with cancer actually die of lung cancer. And throughout my career, I have tried to understand the biology of this disease so that I can, find novel therapies, biomarkers, and also I run a very translational lab, so we want to take our discovery to the clinic. These are some of my disclosures, some relevant to the talk. Okay, so as I said, the Sun Lab at the Icahn School of Medicine at Mount Sinai is a very translational research lab where we study the biology of lung cancer. We study how therapies change the cell intrinsic signaling. We study how it changes the immune microenvironment. We study cellular reprogramming with the idea of how to come up with novel therapies. I'm a molecular biologist and cell biologist by training, so we are very deeply rooted into the research, but we also adapt new technologies, and I'll talk about a lot of that today, of spatial transcriptomics, single-cell sequencing, and also other genomics. And as I wear another hat as a director of the Lung Cancer PDX program, I also collect tissue and blood from patients to make patient-derived xenograft models, which has served as very good model system for studying some of these drugs in the clinic. So for those of you who don't study lung cancer or know as much, so when we think about lung cancer, we think about one disease, but it's really not. Lung cancer is very, very heterogeneous in nature. Every lung cancer patient is actually different from each other. It's a very complex disease, and patients are very unique in terms of the biology they represent, in terms of also the therapeutics that they will respond to, and also the metastatic proclivity of these tumors. But majorly, lung cancer can be divided into these 4 major subtypes: lung adenocarcinoma, squamous cell lung cancer, small cell lung carcinoma, and large cell carcinoma. My lab studies all histologies, all subtypes of lung cancer, but for the interest of time, I only have 45 minutes, so for the interest of time, I'm only going to talk about small cell lung cancer today. So just a little bit of background about small cell lung cancer. Small cell lung cancer is the most aggressive form of lung cancer. It is highly related to a long smoking history, and people are usually detected at a very metastatic stage. And when they are detected, therapies are very limited, so the prognosis is very, very bad. So these patients have close to a year after their diagnosis as a survival. So it's a very aggressive tumor with very high metastatic characteristics. So for the purpose of this talk, I'm going to mainly focus on 2 major projects in my lab, is understanding and overcoming immune evasion in small cell lung cancer, and we have extensively published on that, and also on the molecular drivers and therapeutic targets of lineage plasticity as a mechanism of therapeutic resistance in lung cancer. So these are the 2 major areas that I'm going to focus on today. So again, with small cell lung cancer, it is approximately 15% of lung cancer diagnosis, but in spite of being just 15%, it is the 6th most common cause of cancer-related mortality. Very aggressive disease. It's a high-grade neuroendocrine carcinoma. And about 2/3 of the patients are metastatic at the time of diagnosis. So for the last decade or so, from my postdoctoral fellowship till my faculty appointment, I really invested a lot of time to understand the disease, to understand the biology, to come up with therapies, because therapies are really limited when it comes to small cell lung cancer. Now, just talking about the biology, and this is mainly for the students in the room, so if you think about the genetics of small cell lung cancer, it has a lot of loss of function mutations in major tumor suppressor genes like P53 and RB1. And it also has amplifications in the MYC family members. Now, when we think about therapies which are usually mutationally targeted, like EGFR, KRAS, you may have heard about them, they're usually gain-of-function mutations. So these mutations, these genes or proteins, they're higher, and you design a drug against that. But small cell lung cancer actually represents a very unique challenge. All the mutations that are there in this disease is actually a loss of function mutation. And it is very hard to come up with therapies for loss of function mutations, because you cannot latch onto a mutation and have drug against it. So this has been a major problem with small cell lung cancer. So for that, therapy has been almost limited for the last 40 years. So when a patient comes into the clinic, if you think about the therapy in the United States, when the patient comes into the clinic, they usually get chemotherapy. It's usually a platinum-based chemotherapy with either cisplatin or carboplatin with etoposide, and they get immunotherapy in combination with that. The patients usually respond. They have almost an objective response rate of 60%, But the relapse or the resistance to these drugs are very common. Within months, these patients actually come back with a more aggressive disease. And when that happens, there is really very limited option in the second-line setting for small cell lung cancer. Now, when you think about immunotherapy, and these are some of the trials, that either others have done or we have participated in some of the biomarker studies for these. When you think about the trials, and these are called Kaplan-Meier curves, where you look at the prognosis of patients, either overall survival or progression-free survival. But this is a progression-free survival curve. And if you understand nothing about that, but what you can see is only about 10% of patients actually derive benefit from immunotherapy in the first line for small cell lung cancer. So for the longest time, my goal has been that tail of the curve, which tells you how many patients are responding to that drug. Our goal has been, how do we raise the tail of the curve? Which means, how do we make more patients respond to this therapy? Now, when you think about immunotherapy, some of the characteristics of a cancer that responds to immunotherapy is that if they have high genomic instability, if they have a lot of mutation, or if they have paraneoplastic syndrome. These are pros of why immunotherapy might work in this disease. But some of the problems with small cell lung cancer is that it is also an inherently immunosuppressed disease, which means all the elements that actually makes immunotherapy work, like MHC1, those of you who are studying biology at this level, you may have heard of antigen presentation and MHC biology. So that is inherently suppressed in this disease. What that means, very simply, is that the immune system doesn't know that there is a cancer, so it cannot work against it. Only if the immune system knows there is something foreign will it work against it, right? So if you have antigen presentation suppressed, the immune system doesn't really know that there is anything wrong going on, which means it is a suppressed immune system. So that's why immunotherapy has a very limited benefit in this disease. So my goal has been for the longest time is, how do we make this cold tumor hot? What I mean by cold tumor is that the tumor that is not surrounded by the T cells that can kill the tumors. So small cell is a very immune cold tumor because it doesn't have infiltration of the good T cells or the immune cells that actually work. So when I talked about the tumor suppressor genes like P53 and RB1, the loss of function mutation in these tumor suppressor genes also gives us a tool. Now, if you think about cell cycle, just cell cycle 101, very basic. So cell cycle, and this is school level biology, right? So when you think about cell cycle, there are two major checkpoints in the cell cycle, the G1S checkpoint and the G2M checkpoint. Whenever there is a problem in the replication, the cell stops at these two checkpoints, the G1S and the G2M. It corrects the mistake, and then it carries on with the replication process. This is the biology of normal cells. But we want to take the benefit of having these tumor suppressor losses to actually use the system. And how? If there is a loss of P53, the G1S checkpoint of the cell cycle is actually compromised. And now the entire cell depends upon the G2M checkpoint, which is right here, the G2M checkpoint. It depends upon the G2M checkpoint for any repair of DNA damage. Now we, through targeting these proteins like ATR, CHEK1, V1, ATM, we want to target or take away the G2M checkpoint. Now if you take away both these checkpoints in a cell, now the cell is actually undergoing replication with a lot of error. And enough of these errors, the cell will die because it cannot, there is a threshold of how much error in your DNA a cell can withstand, right? So that's how we want to kill these cells, by taking away the checkpoints so they can cycle and eventually lead to mitotic catastrophe because they have now accumulated a critical load of these errors. And when I joined my postdoc in 2015, my mentor had published this paper in 2012 where she had shown that these proteins that are in the G2M kinases like ATR, V1, and CHEK1, they're actually overexpressed in small cell lung cancer. This was the first indication that this was the case. So since then, we have studied this extensively, and we had shown for the first time that CHEK1, which is a G2M kinase, is actually a vulnerability in small cell lung cancer. We then also showed that another G2M kinase, V1, is another vulnerability. We've launched the first phase 1 clinical trial of a PARP inhibitor, talazoparib, in small cell lung cancer, and since then we have launched an entire field where we have multiple clinical trials of these DNA damage response inhibitors, either as a single agent or in combination with chemotherapy or immunotherapy in small cell lung cancer. Now, this is a work that we published last year, and this work was led by two outstanding postdocs, Dr. Hirokazu Taniguchi from Japan and Dr. Shubhamoy Chakraborty, who is currently in my lab, and he studied in Kolkata. So what they noticed was that this G2M kinase, ATR, is actually overexpressed in small cell lung cancer, which means at baseline, small cell lung cancer has high gene expression of ATR and also protein expression of ATR. Now, when they treated these cells with inhibitors, small molecule inhibitors to ATR called ceralasertib, what they noticed was there was a high degree of micronuclei formation. And I'll walk you through this. I don't know if this is visible from the back. I don't think so. So micronuclei is this DNA-containing structure which is separate from the normal nucleus of the cell. So when you think about a cell, there is the nucleus and you have the DNA. Micronuclei is actually another body which is separate from the normal nucleus, but it contains DNA. What does micronuclei tell you? And if you study virology, you will have an indication of that. What micronuclei tells you is that there is an aberrant chromosomal segregation. Whenever there is an aberrant chromosomal segregation, these micronuclei body forms which contains the DNA. And it also shows that there will be DNA in the cytosol, which is actually abnormal, and it happens during virus infection. So we saw that whenever you treat these cells with an ATR inhibitor, there is actually micronuclei formation and cytosolic DNA formation, which tells you that there may be an immune reaction. Now next we asked that cGAS, which is also now a precursor of an innate immune signaling, we actually stained these micronuclei and we wanted to see the co-localization with cGAS. Now we saw that whenever we are treating with these drugs, which is an ATR inhibitor, there's a lot of micronuclei formation and the cGAS is getting recruited to the site of the micronuclei. And finally, we wanted to see what is the source of this DNA, whether it's the nucleus or it's a mitochondria, because mitochondria also has mitochondrial DNA. For that, we treated our cells with VIVID, which stops mitochondrial leakage, and we saw that the DNA is actually coming from the nucleus, not from the from the mitochondria, because even with VIVID treatment, we are getting a lot of micronuclei formation and cGAS recruitment. So two takeaways from here. Stopping ATR actually leads to micronuclei formation and recruits cGAS to that micronuclei. Now, a little bit about the cGAS STING pathway. It's a very important pathway in the innate immune system. Whenever cGAS is recruited to cytosolic DNA, what happens is that it activates stimulator of interferon genes, or this gene called STING. Whenever STING is activated, it starts an entire cascade of innate immune signaling called the cGAS STING pathway. So STING goes and recruits phosphorylates TBK1, IRF3, and IRF3 goes into the nucleus and starts the transcription of type 1 interferons, interferon alpha or beta. Whenever type 1 interferon gets activated, it recruits or it secretes chemokines like CCL5 and CXCL10. Whenever that happens, you have an active immune response. So that's what the ATR inhibition was doing, is through the innate immune activation, it was launching an anti-tumor immune response. Now, when we treated our cell lines with the ceralasertib or the ATR inhibitor, we saw that the major players of the STING signaling pathway, cGAS, STING, TBK1, they were all getting upregulated and activated. And not just that, we were also seeing the upregulation of the mRNA expression of type 1 interferons like interferon alpha and beta. And we were also seeing upregulation of chemokines like CXCL10 and CCL5. So the entire pathway was actually activated. Now, what we noticed next was that this is great, we are doing it in cell lines, we wanted to now do it in a mouse model. For that, we have a genetically engineered mouse model which has a loss of P53 and RB1, the major alterations in small cell lung cancer. These mice are floxed, so they have a loss, and then we do viral intubation through the intranasal on the intertracheal passage, and these mice actually develop these tumors in the lung. And they're very representative of what we see in patient samples. Now, these mice were actually treated with 4 groups, and if you're not used to seeing these tumor curves, so what this tells you, the growth of the curve is the growth of the tumor in mice. And we gave 4 groups: vehicle, which is just your control, and then we have anti-PD-L1, and then we have ceralasertib, which is an ATR inhibitor, and the combination of immunotherapy with ATR inhibition. And what you notice in both the genetically engineered mouse models is that ceralasertib by itself, which is this blue line right here, it actually regresses tumor significantly. And the combination of PD-L1 with ATR is actually much more effective than immunotherapy by itself. So this combination is actually working in our mouse model. Then we resected these tumors and we did multicolor flow cytometry to look at the immune infiltration. And what we noticed was that we saw an upregulation of CD8 positive T lymphocytes, which are these active T cells which kills your tumor. So we saw that in both our mouse model that we see upregulation of CD8 and upregulation of effector memory T cells. We also confirmed this with immunohistochemistry, where we saw upregulation of infiltrating CD8 positive T cells into the tumor microenvironment. Now, the next question is, and those of you who will go on to do research, you always have to question your data. You always have to double check, triple check to make sure that your hypothesis is correct. So we wanted to see, okay, STING is getting upregulated, but there is a huge difference between correlation and causation, right? Just because something is upregulated doesn't mean it is actually having an effect on the biology. So with the question that we asked is that, okay, STING is activated, but is STING required for this anti-tumor immune response or not? So for that, we deleted STING in our mouse model, and we wanted to see whether the anti-tumor response can actually be reversed. So in this mouse model, what we saw was that when you take away STING, or when you deplete STING, these mice tumor regression is actually reversed. They're no longer getting regressed, which means STING is actually required for this anti-tumor response to happen. Next, so I told you, when a patient comes into the clinic, we treat them with chemoimmunotherapy. Then they get a maintenance immunotherapy regimen with just atezolizumab or durvalumab, the 2 PD-L1 drugs that are approved in the United States. So we wanted to, our goal always is, can we take this drug to the clinic? So the question we asked was that, where in the clinic would this drug be the most effective? So in my mind, I thought, okay, maintenance regimen, where they're getting just this immunotherapy, if we can prolong this phase, then there will no longer be resistance to this drug. So for that, we mimic that in our mouse model. Sorry, I'm a mouse modeler by training, so I always go by default to mouse modeling. So what we did was that we treated our mouse models with this chemoimmunotherapy 4-cycle regimen, or 3-cycle regimen in this case, and then we randomized these mice to receive either immunotherapy by itself, ceralasertib by itself, or the combination. Again, just the takeaway from this is, immunotherapy by itself actually only has a very short interval of response. So the resistant mechanism comes in very easily. But when you treat it with either an ATR inhibitor, you can actually prolong the response in patients. So the resistant emergence can actually be delayed. And this is seen with single-agent ceralasertib, which is this blue line right here, or also the combination with immunotherapy. So now our goal is to actually do clinical trials with ATR inhibition in combination with immunotherapy in the maintenance setting. Now, interestingly, what we noticed in our mouse model, if you remember, I said that small cell lung cancer actually has a suppressed antigen presentation because it has low MHC1. Now, what we saw was that decreasing ATR can increase MHC1 expression, which means it can also activate antigen presentation in small cell lung cancer. And we saw this in our cell lines, in our mouse model, so we are pretty sure at this point that DDR, or DNA damage response inhibition, can also activate antigen presentation in small cell lung cancer. So we always want to test whatever we find in the lab in the clinical samples. For that, in this clinical trial of berzosertib, which is an ATR inhibitor in combination with immunotopotecan, we collected these cases pre-treatment and post-treatment. We did RNA sequencing to look at the gene expression, and what we noticed was that even in the clinical samples, we are getting an upregulation of MHC1 and MHC2. So this was now a validation of our research in the lab that even in patients, MHC1 and MHC2 was getting upregulated. So this is sort of a summary of this story and DDR as a whole. So what I call it is a marriage of checkpoints. You have the cell cycle checkpoint and you have the immune checkpoints. If you combine these two, when you target any of the DDRs, I spoke about ATR, but we have also published quite a bit on CHEK1, on PARP. So when we target these DNA damage response proteins, we launch an innate immune signaling activation through the SIGA-STING pathway. And when this immune activation pathway is launched, it leads to this cascade of signaling which touches everything. Antigen presentation, increase of HLA, MHC1, macrophages, M1 and M2 macrophages. It changes immunological cell death. So there's really a blanket of immune response that is launched. And what that leads to is a regression of tumor in mice, but not just in mice, because we are not, we want to take this to patients. This is also now a clinical trial in patients. We've published quite a bit on this. So this is a study with PARP and CHEK1. This is a study with PARP again, and this is a study with V1. And now we have about 14 clinical trials that we have launched with DNA damage response inhibitors in combination with immunotherapy for small cell lung cancer. These are mainly maintenance and second-line clinical trials that we are doing right now. So I'll switch gears a little bit. I'm happy to take questions from anyone in the audience at the end, but I'm going to switch gears a little bit and talk about the second project. So one of the problems with cancer therapy is that the drugs that are currently in the clinic does not work, right? They work, the patients respond for a very short period, and then we get resistance to those drugs. And when the disease comes back, it's actually more aggressive than the initial tumor. And from that point, the patient has very low survival rate, right? So what my lab really is trying to understand is how do these resistance evolve either to tyrosine kinase inhibitors or to immunotherapy or to chemotherapy. And this is a story that I'll talk about, which is a very unique phenomena that the cancer does. It's called lineage plasticity. So when you think about non-small cell lung cancer, it is very different from small cell lung cancer. So in non-small cell lung cancer, there are major alterations or genetic alterations which are gain-of-function mutations like KRAS, P53, not KRAS, ALK, EGFR, others, right? And now there are drugs that are against KRAS, against EGFR, against ALK, ROS. These are all the genes that are actually mutated in non-small cell lung cancer. And thanks to molecular testing, we are doing really well in the first line for these patients. We have drugs for everybody, almost everybody, for all of these genetic alterations. But the problem is, again, resistance. About 60% to 70% of these patients will only have a very short response duration to these drugs. They will eventually develop resistance, and when they do, it's not a good prognosis for these patients. So resistance can develop. Those of you who are studying signaling in your classes, resistance can develop in many different ways. If you're targeting, let's say, an EGFR pathway, resistance can develop through a secondary mutation in the same pathway or through targeting other pathways, the oncogenic signaling like MAPK signaling and other signaling, RAS signaling, JAK-STAT signaling that get turned on. We are interested in all those, but what my lab majorly studies is this process called histological transformation. And just for the audience, you may not be aware of this if you don't see it in the clinic every day, is when a lung adenocarcinoma patient is treated with a tyrosine kinase inhibitor, after a duration of time, they change to small cell lung cancer or squamous cell lung cancer. What that means is that you're not even treating the same disease anymore. The disease has changed. It is no longer lung adenocarcinoma. It is now a small cell lung cancer. So no wonder the therapy that is for lung adenocarcinoma doesn't work anymore, right? So it's a no-brainer to everybody. So we've studied this phenomena. We want to understand the molecular mechanism. We want to come up with drugs to treat this transformed tumors. So again, going back to developmental biology, everything that you study in cancer, you will study in normal biology. So those of you in your developmental biology classes, you have come across these terms called dedifferentiation, transdifferentiation, right? This is a normal biology of cells. This is how a body develops. Wound healing, everything happens through these normal processes of these cells. But cancer is actually a very intelligent system, right? So they actually hijack these processes in order to become more aggressive, more resistant to therapy. So they hijack these processes of transdifferentiation, dedifferentiation, lineage plasticity. So much literature has been published in prostate and pancreatic cancer that in the recent iteration of hallmarks of cancer, we see unlocking phenotypic plasticity and non-mutational epigenetic reprogramming are actually hallmarks of cancer. So in an EGFR, the cancer that we study in this context, when an EGFR mutant patient actually comes into the clinic, they're treated with tyrosine kinase inhibitor like osimertinib. About 14 to 20% of these cases actually transform to small cell lung cancer, so it's quite a bit. And not just tyrosine kinase inhibitor, small cell lung cancer transformation can happen as a result of immunotherapy, and not just EGFR, it can happen in ALK, it can happen in KRAS, right? So it's an all-pervasive phenomena, and it happens in about 20% of patients. So this is actually really alarming. So what are the characteristics of these tumors? They are very much lung adeno at the beginning, so they express all the lung adeno markers, EGFR signaling and everything. And they do not have any neuroendocrine marker, which are the characteristics marker of small cell lung cancer. But when they transform, they are now expressing neuroendocrine markers. So I think you will appreciate this. This is actually changing from this MAPK-driven lung adeno to this P53 RB1-driven, very aggressive small cell lung cancer. Now, to understand the biology of these tumors, what we did was that we identified mixed histology cases. What I mean by mixed histology, that in the clinic, both the lung adeno and the small cell lung cancer actually exist in the same histological space. I don't know if there is any pathologist in the room, so really, when you look at the slide, you can see both these two tumors in the same histological portion. We identified these cases, we microdissected these samples, and we did multi-omic sequencing with whole exome sequencing, RNA sequencing, methylation analysis. So what we noticed was that this is just an oncoprint that you see from our whole exome sequencing, and I know no one at the back can read what I have written there. But what I'm going to tell you is that all the mutations that are present in the lung adenocarcinoma are retained in small cell lung cancer, right? So the mutation doesn't change. It's the same cancer, just changing its phenotype. And the major mutations that we saw come up was P53 and RB1. They were highly mutated in the tumors that actually transformed to small cell lung cancer. And this is showing some of the major mutations, UCP53RB1, but there are other mutations that we are seeing like KMT2B, NFE2L2. These are major players in epigenetic regulation, not family regulation, and we are trying to understand whether these could be potential drivers of lineage plasticity or not. Now, our question was, are there non-mutational cues to this process of lineage plasticity? For that, we looked at the RNA sequencing data and also to the methylation data. Dr. Dasgupta, how much time do I have? Are you looking at the time? 10 minutes? Okay, I'll finish. So when we looked at the principal component analysis, what we noticed was that with gene expression analysis and with methylation analysis, again, if you're not familiar, this may not make a lot of sense to you. So anything that is closer to each other in this principal component analysis are very similar to each other in the genetic mechanism or genetic programming, right? So if you see here, the transformed small cell lung cancer and the pre-transformation lung adenocarcinoma, they're actually very similar to each other at the RNA level. And they are a middle stage to classical lung adeno, classical small cell. At the methylation level, something really interesting came up. What we noticed was that this transformed small cell has a huge degree of overlap with the pre-transformation lung adeno, and epigenetically they're very different from classical small cell, which means the take-home for the audience is that transcriptionally, this mixed histology state is a state between classical lung adeno and classical small cell, and epigenetically, it actually retains the epigenetic memory of the pre-transformation lung adenocarcinoma state, and they're epigenetically very different from classical small cell. They look like small cell, they're treated like small cell, but they're not really small cell, which means the therapies that you are giving it, the patients don't usually respond to these. So we looked deeper into the gene expression programs. The gene expression programs that came up very, very clearly was that there is an upregulation of cell cycle, DNA repair, epithelial to mesenchymal transitions, PRC2 complex, Wnt signaling, and Notch inhibition. But interestingly enough, the signaling that is actually suppressed is immune. So immune evasion or immune suppression is actually a permission for lineage plasticity or histological transformation to happen. Now when we looked at the cases, the lung adenocarcinoma tumors that are never transformed, so there are 80% of the cases that never transform to small cell, right? Only 20% of the cases do. So we compared that 80% to this 20%. And what we noticed was that the signaling pathways like cell cycle, DNA repair, PRC2 and AKT differentiate the tumors that transform and the ones that don't. So these are potential drivers of lineage plasticity. But interestingly enough, again, the pathways that were suppressed in these tumors was, again, anti-tumor immunity. So you need to suppress the immune microenvironment for this process of histological transformation to happen. So we also now confirmed that with multiplex ion beam imaging, where we can image up to 40 markers in a histology slide. If you cannot see anything from the back, hopefully you can see that there are no colored dots in this space. There are no colored dots, which means there are no immune cells in this transformed small cell portion of the mixed histology tissue. So immune evasion has to happen for the process of histological transformation. We did a lot of imaging with Orion platform, with multiplex ion beam imaging, and this may not make a lot of sense, but what you can notice is that immunologically they're very heterogeneous, which means these tumors are very different from each other in terms of immune status or immune microenvironment landscape, which again presents a unique challenge in the clinic because they are not immunologically same to each other. And there is a suppression of CD8, CD4 in the transformed tumors. Now, after that, we wanted to look at the trajectory, which means, can we apply single-cell sequencing to see whether small cell actually develops from non-small cell lung cancer? And for that, we used an in-house method called pseudotime. So what pseudotime does is that it puts genes in the time scale on which it occurs, right? So if you see here, non-small cell is an earlier event and small cell is a later event in this process of pseudotime. And when we also looked at individual genes that either go up or are suppressed, we see an upregulation of several genes like ACL1. We also see an upregulation of Notch inhibitory ligand, DLL3, in this process of transformation. And we see the suppression of Notch ligand like JAK2 or Notch itself to be suppressed. And I'll summarize my talk in a few slides. You will understand why I'm showing you this slide. Now then, we applied other single cell techniques to look at, because our really goal is to see what is the trajectory. You know the beginning and the end, non-small cell to small cell, but we don't really know what road the tumor takes. We don't know the path. I came from South Calcutta, I came to Belvermont. My destination and my origin is fixed, but I can take 10 different routes to get there. So what we really need to understand is which route does a tumor take to have this process of transformation. And that's what we did with the single cell sequencing. So when we did this, what we call the latent time analysis, what we noticed was that, and this is each cell that is arranged according to that. Now these tumors, they are very intelligent. They actually turn on all these lung adenocarcinoma markers in the beginning. And when they actually transform, they're phasing out these genes, and it's a relay race. They're phasing out the lung adenocarcinoma genes, and the small cell genes are taking over. So you will see here, all the lung adenocarcinoma genes are very high in the beginning. They get lower, 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 and you see the upregulation of all these genes that happen very highly in small cell lung cancer. So it takes a very specific route. We are currently doing a lot of work in the lab to understand this route of lineage plasticity, but it's very complex and it will be very patient specific. Now when we applied with the de novo small cell lung cancer and transformed small cell lung cancer, what you can notice from here is that I hope you can appreciate that there's a lot more color and a lot more spread in this particular graph. What that means is that these tumors are very different from each other. The more space they have in this UMAP, the more different are they from each other. And the more the colors, the more the genes they're turning on. So they're very heterogeneous from each other, and this is intertumoral heterogeneity. So the heterogeneity between one tumor and another. The next is, does this happen even within one tumor? Again, this, if you notice here, there are a lot more colors within the same space, right? So here you see predominantly red, predominantly blue, and predominantly dark blue and light blue. But here, even within one tumor, there is purple, yellow, red. So even within one tumor, there's so many gene expression programs that are working. And so these tumors are very, very heterogeneous. Finally, can we track? So for the clinicians in the room, it's a real problem, right? How do you predict when this transformation is going to happen? So we asked, can we track lineage plasticity? And you can. We can use blood. We can use blood draws from the patient. We can have epigenetic sort of these programming that happens. So we do cfDNA sequencing, fragmentomics, to look at this, these predictors of transformation. And we are not there yet. Tissue is still our gold standard, but I think within a couple of years, we will be able to come up with markers in the blood that will be able to predict this process of histological transformation. So what are the therapeutic targets? My lab is all about finding therapeutic targets, right? So if you remember from the heat map, even if you don't remember, AKT signaling was one of the signaling pathways that were going up during this process of transformation. So we treated our patient-derived xenograft models with samatolisib, which is a PI3K/AKT inhibitor, in combination with osimertinib, which is a tyrosine kinase inhibitor for EGFR. What we noticed was that the treatment with samatolisib along with osimertinib is actually way more effective than osimertinib by itself. So AKT could be a therapeutic target. The next target, so when we looked at these tumors, DLL3, which is called the delta-like ligand 3, which is an inhibitory Notch ligand, if you think about the Notch signaling, this particular protein is really high. 93% of these tumors actually express DLL3. So now there are bispecific T-cell engagers for DLL3 targeted therapies, and we are actually doing clinical trials for this DLL3 targeted therapy in this transformed small cell. Finally, we study lurbinectedin a lot. Lurbinectedin is like a chemotherapy. It is approved in the second-line setting for small cell lung cancer. It is a DNA minor groove inhibitor. And when we treated our mouse model with lurbinectedin in combination with osimertinib, we saw that it is actually very, very effective. This red line is very different from the yellow line and the black line here. So the combination of lurbinectedin with osimertinib is very effective, which tells you that this drug will be effective. So we have started a clinical trial, the first in-human clinical trial combining lurbinectedin with osimertinib in transformed small cell lung cancer, which is called the LOT study, and I'm a co-PI of this study. So I want to summarize. This process of lineage plasticity is very complex and it's very, very dynamic. It doesn't happen in a day. It takes months to years for this process to happen. It goes from the lung adenocarcinoma state to this mixed histology state, which I talked about quite a bit. What do we understand about the genetic alterations that can be predictive? P53 and RB1 loss are the patients who are at very high risk for this process of transformation. So we know that. We don't know much more. What are the transcriptomic predictors? We see upregulation of several transcription factors. You will see 3 names mentioned here: PAU3F2, 1CUT2, and FOXN4. These are all lineage determining transcription factors that we know from other cancer like prostate, pancreatic. We also see suppression of Notch signaling, and Notch has a very important role to play in neuroendocrine differentiation. So we see a suppression of Notch signaling. When these tumors actually transform, they're very different from each other. They're very heterogeneous in nature. We see an upregulation of several targetable genes like AKT family members, DNA damage response, and other genes. We are investigating that in the lab. They could be potential therapeutic targets. And very importantly, we see an immune suppression throughout this process of histological transformation. We don't know a lot. I'll be very humble in accepting we don't understand almost 80% of what is going on here. And these are the question marks that I have here. What are the other genetic alterations? We don't know. What are the other transcription factors? We don't know. What makes these tumors decide which kind of small cell lung cancer they will be? We don't know. How do they shift from one another? We don't know. You must be thinking, she doesn't know much. But that's the problem of biology. The more you study something, the more you start to appreciate how much you actually don't know about the complex system. And to do anything well, you have to do it yourself. So we are now starting a clinical trial where we are going to enroll all these patients with KRAS, ALK, EGFR, and we are going to track them. Every patient with these genetic alterations, if they also have a co-alteration in P53 and RB1, we are going to track these cases. We are going to collect blood, we are going to collect tissue, and we are going to make cell lines, PDXs, and we're also going to do single cell from every time point. What is the goal? The goal is we want to understand what drives lineage plasticity, what is the route that these tumors take, and finally, can we have therapeutic options for these cases? With that, I'll actually stop here. I want to thank my entire team. I have a fantastic team there. They're amazing. I actually have Abhishek's name still here because believe it or not, we are still writing papers together. So never let go of your mentors because they will be with you for the rest of your life. So please respect your mentor and be with them forever. Abhishek and I are still writing papers together. And I want to thank all my collaborators, not just in Mount Sinai, but all over the world actually. I want to thank my funding agencies. Without funding, nothing is possible. And finally, patients and their families who are incredibly generous to us with the tissue, with the blood, with everything. With that, again, I'm really humbled to be here, and thank you so much for listening to me. I know it's a lot, but I'm happy to take any questions from anyone in the audience. Thank you. Thank you very much.
Thank you, Dr. Shen, for your excellent talk. It's a high level of research. Lung cancer is nowadays a very burning issue in our society, also in India. So new insight regarding the therapeutic approach. I'll be happy one or two questions from the audience, especially the students.
No questions? Oh.
And
Can we pass?
Adenocarcinoma to small cell carcinoma, it takes more time to transform. Then why it is difficult to track the transformation?
That's a great question actually, because you pose a very important problem in the clinic. So when these patients come, right, they're detected with lung adenocarcinoma, and they transform over the period of, let's say, 2 years, 2.5 years, right? But you don't really know who are going to transform. So you don't collect a biopsy from a patient unless you really need to, unless they relapse. By the time they relapse, they're already small cell lung cancer, right? So it was hard to track these cases in the earlier setup. But now with the trial that I showed you in the last slide, we're going to collect blood with very, not very frequent, but within a few months, especially for these patients who have a P53RB loss. We are going to diligently track them because they are high-risk population. So we feel like in a few years, we will be able to track this process of lineage plasticity. Till now, we were limited with the tissue that we were getting. It's very hard. It's actually, the clinicians won't take out chunks of tissue from a patient unless they need to, right? It's not a part of the clinical practice. So that's why we were unable to get these middle time points. But now, with blood draws, if we can have a biomarker in the blood, it's a much easier ask for patients to be able to draw 1 vial of blood from them. So now, I think going forward, we will be able to track it. That's our hope.
Okay, thank you. And another one.
Hello, this is Dr. Kaustav Chakraborty, Assistant Professor Geology, Barashat Government College.
Hi.
Thank you, Madam, for such a wonderful and interesting presentation. I have a couple of queries. You said that there is a problem of immune suppressions in most of the cancer patients. And you're talking that, that there is an antigen presentation is hampered. So, as far as I know, that dendritic cell-based immunotherapy or DC vaccine is an emerging concept in the Western world. So whether it is applicable in your topic, that is the non-small cell lung cancer?
Great question. So what we are doing now, since MHC is actually suppressed, so as I said, it's an immunosuppression, so now we are using newer immunotherapy approaches like BiTEs, so bispecific T-cell engagers, which has, what I mean by bispecific is that it has a CD3 component and it has a marker in the tumor cell, so DLL3. So what it does is it takes away all the problem of antigen presentation. It's pulling CD3 immune cell and it's pulling DLL3, which is expressed on the tumor cell, and it's making it meet. So now, instead of going the immune checkpoint route with PD-1, PD-L1, CTLA-4, we are using these newer kinds of immunotherapy like bispecific T-cell engagers, CAR-Ts, to then treat these tumors. And actually, tarlatamab, which is a DLL3 targeted bispecific T-cell engager, is working fantastic in these patients. For the first time, we're actually seeing long-term responders in the second-line setting. And these are hard drugs, they're costly, so adapting that in a setting outside the United States may be a challenge. And they're also quite toxic because you get immunological toxicity in these ICONs and IRSs with these tumors. So they're not the easiest of drugs, but for the patients that they work in, it's really a blessing.
Thank you, madam. My second question is that the small cell cancer also has the capability to metastasize, I think.
Oh, very,
okay.
very.
So do you have checked any cancer stem cells marker by immunohistochemistry or any by flow cytometry after taking single cell suspension from lung tissue?
Yeah. So we see a lot of SOX family members, especially when they metastasize. So we published the first single cell atlas for small cell lung cancer. And what we noticed was that any tumor that we are getting from the metastatic side, either in the brain or in the liver, they have a high stem cell-like signature, so which means SOX family members are really high, SOX2, SOX9, they're really high. So yes, metastatic tumors, they have a stem-like signature that they're turning on in order to metastasize. We are seeing that.
Okay, thank you very many thanks.
Thank
you, Dr. Shen.
Thank you.
Our next speaker, Dr. Argo Chattopadhyay. It is my absolute privilege to introduce to you a highly respected name in the field of clinical immunology and rheumatology, Dr. Argo Chattopadhyay, a renowned rheumatologist based in Kolkata. Dr. Chattopadhyay brings with him a wealth of experience and a stellar academic background. He completed his MBBS and MD and went on to pursue his DM in clinical immunology and rheumatology from the prestigious Postgraduate Institute of Medical Education and Research, Chandigarh, one of the India's finest medical institutions. Over the years, Dr. Chattopadhyay has established himself as a trusted authority in managing a wide spectrum of autoimmune and rheumatological disorder, including systemic lupus erythematosus, rheumatoid arthritis, scleroderma, spondylitis, myositis, and vasculititis, and a host of rare primary immunodeficiency and autoimmune conditions. What truly sets Dr. Chattopadhyay apart is not only his clinical expertise and grasp over the latest diagnostic and therapeutic technology, but also his deeply compassionate, patient-centric approach, known for his gentle demeanor, methodological case evaluation, and unwavering commitment to each individual he treats. Dr. Chattopadhyay embodies the ideal of the physician who heals both body and soul. Today we are honored to have him and with us to share his insights and experience in this highly specialized and evolving field. I am confident that his talk will be both enlightening and impactful for all of us. Please, Dr. Chattopadhyay.
So, very good. And it's definitely a proud privilege for me to be here at Belur Vidya Mandir. So, just before starting my talk, I just want to know how many of you don't understand Bengali here? Anyone here who don't understand Bengali?
Almost all. Bengali.
Don't understand Bengali. Because I have a fear if I speak in English, then I may use more medical term that may not be useful for you. So, you have been through an extensive journey of lung cancer, all its ups and downs and too many cytokines and chemokines and genetics involved in a specific type of lung cancer. But today, my topic is autoimmunity and how autoimmunity immunity can translate into autoimmunity, and autoimmunity, how can it translate into our practical treatment and therapeutics. Anyone in the audience can tell what is this picture about? Anyone? No. So this is a smallpox picture, and the entire discipline of immunology and autoimmunity, this immunology discipline, grew out of observation. The Latin term immunis, that means exempt. From that, the English coinage of immunity, and that means that giving protection against infections and other foreign elements that comes. So in 430 BC, for the first time, there is historic description when in the plague in Athens, only those who had recovered from the plague could nurse the sick because they would not contact the disease a second time. So this was the first possible description when the concept of immunity was described in history. But unfortunately, it took another 2000 years to understand and to convert that successfully into medically effective practice of this observation
into an immunology or clinical immunology setting. So if we talk about immunity and autoimmunity, before moving into autoimmunity, you need to understand what is immunity. So in a broad way, we have two separate areas of our immune system. What is an adaptive immune system and humoral and cellular. These two major immune system that plays the all roles starting from your disease, including cancer. So everywhere there is a disease, there is some kind of has to be some kind of immune aberration that leads to development of some kind of disease. So what is all about humoral immune response and cell-mediated response? So for the sake of ease of discussion, let's skip the immune system cells. There are n number of cells, but two major cells in the immune system, one is B cell, another is T cell. So B cell, which are responsible for development of autoantibodies. So B cell are responsible for antibody development. So whenever something foreign comes into our body, B cell get activated, they produce some antibody, they combine with the antigen and they eliminate that thing from the body. So this is the way the B cell works. On the other hand, T cell how does? So they are more sophisticated and they works in a different way. They don't just have a knee-jerk reaction like just seeing someone, they start producing something. So they have different levels. They give some signals, they produce some chemicals. These chemicals in turn activate both B cells and another T cells. And some of these T cells directly kill the entire cell of the pathogenic, which contains the pathogens. So they both way activate the B cell for development of the antibodies, and on the other way they activate the T cell, another subset of T cell, which helps in killing the cells which already phagocytosed the pathogen. Anyone having any idea of this picture? So this is a Greek mythology, though the concept is there is a continuous cycle of life. So one end is starting and another is ending, but possibly this is one of the historic description of autoimmunity, that one is eating self. So self-destruction is the autoimmunity. So what is the basis of autoimmunity? For understanding the autoimmunity, we need to understand immunological tolerance. Like that, whatever we are seeing in the external environment, exactly same thing is happening to your body. So what is the tolerance? So whenever, see, every time body is trained to have to look for the abnormal signals. Whenever there is abnormal signals, these T cells get activated, B cells get activated, and they try to destroy the cells which are inside the body, because they have been trained in that way. Unless you destroy them, they will destroy the body. So, but sometimes the body, there are some abnormal protein production which are actually not from outside, but from the self. So if there is immunogenic antigens like from the outside, microbes or something, the antigen presenting cell that engulf the thing and represent to that T cell, and T cell get activated, and upon activation they stimulate the B cell and T cell and then destroy the whatever immunogenic antigens is there. But if it is from inside the body, then we call it tolerogenic. So whenever there is something inside the body can understand. If they can understand it is inside the body, then there is a second type of mechanism where they do not react, they do not produce the effector cells to destroy the cells. So this is called tolerance. So that is practical and functional inactivations or cell death or sensitive to suppression. So if I appreciate my T cell or your T cell appreciate that something, the signals or whatever is getting
noticed by the antigen presenting cell is from the body, then they don't react in that way, so they suppress the subsequent pathway of reactions, and that is the tolerance. So there are two types of thing, one is immunogen and another is tolerogen. So antigens that induce tolerance are called tolerogens, the self-peptides rather than the immunogens which are external thing. The same chemical compound can be both as immunogen and tolerogen depending on how and where it is present in the immune system. So this is the basic of autoimmunity. So presence of specific antigen ignored by the lymphocyte, that is called immunological ignorance, and that happens many a time in a cancer thing, that there are abnormal cells within the body, but body cannot appreciate those abnormal cells because they are smart enough to get masked by them. They're smart enough to get escaped from the antigen presenting cell in that way. That's why there is a necessity of upregulation of the T receptor cell in the cancer cells. So this is the immunological ignorance. So there are two separate types of tolerance. One is called central tolerance, another is peripheral tolerance. What is central tolerance? Central tolerance occurs in the central generative lymphoid organs, thymus, bone marrow. These are the area where the central tolerance occurs. Vis-a-vis where the peripheral tolerance in the peripheral tissue. So it's a two-step mechanism by which body is trying to prevent self-harm. How? So this is the balance between the activation of the effector T cells and regulated T cells. So if there is activation against the self, then it will be an inflammatory response. If there is no activation upon recognition of the self antigen, then it's in tolerance. What about the central tolerance? What are the mechanisms of central tolerance? So death of the immature T cells, a generation of the CD4 regulated T cells. So this is one way called negative selection, and another way of development of the regulated T cell. So if any of the T cell is exposed to the antigen presenting cell and body is appreciating that this is too strong stimulus, then body try to eliminate them by apoptosis, by cell death. Another way, body can learn that these are the signals, these are actually from within the body, not from the outside. So I need to learn this, and with this learning, they developed regulated T cells. These regulated T cells on exposure to same kind of antigens, they will not react any kind of this kind, they are not engage any kind of T cells or any kind of immune response, because they have already learned this, that these kind of response are coming within the body. So these are the thymus is one of the major area where self antigens are seen, and they have been training with this, that the central tolerance part of both the mechanism has been there in the thymus. There is something called autoimmune regulator gene. So what happens, this regulator gene actually regulates the tissue restricted antigens on exposure to APC. Ideally, if it is a self-reactive T cell, then there will be apoptosis. So that's called negative selection or deletion of the thing. But that somewhere is one of the genes, is AR gene, which is responsible for secreting these tissue restricted antigen. And if there is a mutation of this gene, then there will be no further restriction of this self-reactive T cell, and they will keep proliferating, and they will subsequently do tissue injury and autoimmunity. So what is the clinical consequence of AR gene mutation? There is a disease called autoimmune polyendocrinopathy with candidiasis and ectodermal dysplasia. EPSID, there is a disease when there is a mutation of this gene. So this is an autoimmune disease when the pathway of inhalation gets lost, body develops autoimmunity. When we look into the mouse model, it produces lots of autoantibodies in multiple endocrine organs, including retinas. And endocrine organs in this particular model is the major target for autoimmune attacks. Now come to the peripheral tolerance. So this is a two-step check model. If somehow the central tolerance unable to recognize the thing and they come into the peripheral circulation, then there are three methods of making the peripheral tolerance, or one is energy. Energy, what is energy? This is functional unresponsiveness. So whenever there is a binding of the autoreactive T cell to MHC, what they cause, they inhibit, they produce some inhibitory signals, and that cell remain non-functional or unresponsive. So this is called energy. Suppression. So we have learned from the central tolerance. When central tolerance has learned something, they send the regulatory cell to the peripheral circulation from the previous exposure. This regulatory T cell, subsequently, when there is a binding of the autoreactive T cell, they block subsequent activation. And third one is deletion, same as the central tolerance. If you cannot control, if you cannot suppress, then you just delete the thing, because otherwise it will be harmful to the body. So regulatory T cell inhibits self-reactive T cell. So in thymocytes, if low affinity for self-antigen, then that can be inhibited by the Treg cells. If high affinity, then just straightaway apoptosis. Intermediate affinity for the self-antigen, upregulation of the FOXP3, and they make it a Treg cell, which in turn take care of the both central and peripheral tolerance. There is something called peripheral tolerance known as link suppression, because this Treg cell, if this cell has affinity for peptide A and peptide B, it has a potential to inhibit both the thing. So there is a link suppression and subsequent inactivation of the autoreactive T cell. So energy will, this is like again recognition of the self-antigen, then signal blocking. This is the main thing, is the signal blocking, and that in turn become unresponsive to T cell by the engagement of the inhibitory peptides. And the major area of the whether energy or activation will be there, the presence or absence of the co-stimulation is the major determinant whether cell will move into energy or activation and proliferation. Now, you have heard lots of PD-L1 in the previous lecture. So, what is that? So, these are the T cell receptors and the ligands for APCs. So, whenever there is B7 and T cell receptor CD28, there is activation of the naive T cells. Whenever there is combination of the CTLA4, there is inhibition, that it mediates suppressive function of the regulatory T cells. On the other hand, ICOS, this activation of the helper T cell and antibody response. And PDL and PD1, it causes inhibition. What happens in real life? So this is the, again, recapitulation of, so PD1 is, if there is absence, CTLA4, there is a proliferation, and with CTLA4, there will be suppression, so functional inactivation. This is the one of the pathway of the energy. So CTLA4 completely inhibits B7 and CD28 engagement. So these are the normal body defense mechanism. But what happened to the cancer cell? What happened to the cancer cell when there is an activation? So we want to more and more activation of PD1, because cancer cells are getting escaped from the pathway. So whenever with the therapeutics we are trying to block PD1, then the other way problem, they are developing a sets of autoimmune disease. So this kind of immunotherapy, PD1 inhibitors, on the one way, they are helping in managing different kind of malignancy and cancer, but as because they are also very important for the central tolerance, they are on the other way, they are developing
series of different kinds of autoimmune features. These are the, and nowadays, whenever they are planning for starting this immunotherapy involving the PD-1, so they have usually before starting the immunotherapy, they have a panel including a rheumatologist, because most of the patients develop different kinds of autoimmune manifestation, including very severe arthritis, muscle involvement called myositis, vasculitis, and all different sets of autoimmune disease when these group of patients are being treated with this kind of PD-1 inhibitors. So these are two major, CTLA-4 and PD-1. So CTLA-4 is a major site of action in lymphoid organs, while PD-1 is the peripheral tissue. This is induction in the effector phase, the CD28 here, the chronic antigen receptor stimulation. So inflammatory reactions following antibody treatments are more severe with the CTLA-4 than the PD-1 inhibitor. So these are the challenge. On the other hand, whenever you are trying to treating a disease with some group of therapeutic agents, on the other way, they are opening the other arena of autoimmunity, because these are the normal component of the immune system and they are helping in your central and peripheral tolerance. So this is what I am telling. So lots of inflammatory disorder including colitis, severe adverse events. So before starting, we need to check for whether the patient is already susceptible for any other autoimmune disease, or during the therapy even there are cases where we need to discontinue therapeutics targeting PD-1 and CTLA-4 because of the very severe kind of and life-threatening autoimmune response to the agents. So now come to the B lymphocyte tolerance. The self-polysaccharides, lipids, and nucleic acids are the T-independent antigens, are not recognized by the T cells. So they usually escapes from the T cell tolerance. Again, B cell also has in both central and peripheral tolerance, and almost the same kind of mechanism, but B cell has antibody, so they have some kind of a specific phenomenon called receptor editing. So when self-antigen recognition is there, self-reactivation of the B cell, there is a method called receptor editing, expression of the new immunoglobulin IgG in the V region, so that on receptor editing, they can become a non-self-reactive B cell. Next, the apoptosis, the deletion, the way the T cell occurs, and energy is another thing, reduce receptor expression and signaling, they become a practically non-functioning B cell to the self-antigen. In the same way, the peripheral B cell lymphocyte tolerance is functional activation, energy, deletion, and regulatory by inhibitory receptors. These are the major pathway of B cell peripheral tolerance. What about the tolerance of commensal microbes and fetal antigen? So we all know we harbors millions and millions of the commensal bacteria in our skin, in our GI tract, but we don't react to them. And the largest example of fetus within a mother womb that they carry despite being in separate MHC expression. So what about the microbes? Abundance of the IL-10 production by regulatory T cells, an unusual property of dendritic cells such as signaling from the TLS leads to inhibition rather than activation. And separation of the some bacteria from the intestinal immune system by the epithelium is another natural way of protecting the thing, that they don't get exposed to the immune system directly and they can survive in the intestine. But the mechanism of tolerance to the commensal bacteria in the skin is still not very well-known thing. So does the fetal antigen, thus exclusion of the inflammatory cells from the pregnant uterus, poor antigen representation to the placenta, inhibitor to the greater harmful Th1 response to the healthy pregnant uterus. So these are the mechanisms where body protects some of the beneficial thing for the benefit of the own body. Now comes to the autoimmunity. So whenever there is a lack of central tolerance, break of the central tolerance or the peripheral tolerance, there will be development of autoimmunity. For the development of autoimmunity, we need some genetic susceptibility or reaction to the environmental stimuli in a setting of the genetic susceptibility and environmental stimuli. The failure of the self-tolerance is the key for the development of the whole set of autoimmune disease. What are the genetic susceptibility? So these are some MHC alleles which are strongly linked with different autoimmune disease, like for ankylosing spondylitis, HLA-B27 has been shown to be with a relative risk of 90. Like that for rheumatoid arthritis, HLA-DRB01, 04. For diabetes, HLA-DRB1, 0301. Pemphigus. So these are the genetic factor, predisposing factor for the development of the autoimmunity. So the possible mechanism, self-tolerance, the loss of self-tolerance. So this is the normal protection. If there is a loss of self-tolerance, induction of the co-stimulatory APC, so when on activation there will be autoimmunity. Other way, there is a molecular mimicry, and this happens most with the viral infection. Every year we see patients with, during the puja time, there is a fever and lots of patients develop joint pain following on fever. This is a classical example of molecular mimicry, because the viral infection leads some kind of molecular mimicry, and body cannot differentiate between the viral proteins and the joint proteins, and they start attacking the joints as a bystander of immune activation and self-harm and autoimmunity. So these are huge, there are huge list of genes which are under research, but none of these are monogenic disease, and these genes are most of the time is a predisposing or background genetic thing which is required for the development of the subsequent autoimmunity. But there are some genes which are as a single mutation can cause autoimmunity, like the ARVI we have already discussed, CTLA4, FOXP3 and FAS. These are the genes which single gene mutation can lead to autoimmunity by different kind of subsequent cytokine and chemokine expression. Moving to a translational part, that this concept of autoimmunity can be translated into our daily practice. I will tell 3 different diseases of classical example. One is rheumatoid arthritis. That's a classical example of joint dominant disease and arthritis. So rheumatoid arthritis is a joint dominant systemic disease, though that can affect your eyes, nerves, liver, skin, bones, kidney and heart, but still it's a joint dominant disease. So there is lots of inflammatory reactions in the joint. And if we look in the pathogenesis of the rheumatoid arthritis, in every step we have some options where we can intervene. So in practically, like we use in our day-to-day practice, anti-TNF like adalimumab, etanercept, these are for the therapeutic. But there are is an interesting story of the IL-1 inhibitor. This is one of the cytokines discovered in the early phase of the rheumatoid, and when the in vivo research, the concept was there will be cure for the rheumatoid. But there is a lots of difference between in vivo and in vitro experiment. So when we practically, the clinical trial started with IL-1 inhibitors for a setting of the rheumatoid, there is absolutely no response to the IL-1 inhibitor. These molecules are now used for some of the autoinflammatory syndromes, but absolutely not very useful for rheumatoid arthritis. Like that, in the different, like every step we have some options, and most of these are available as our day-to-day clinical practice. Like IL-6 inhibitors, we have IL-17 inhibitors, we have CD20 inhibitors. So these are the molecules we use in our day-to-day clinical practice. So the only way forward, if we know the better pathogenesis, we can have better molecules to treat the disease. So these are the chemokines and cytokines pathway. So these are the main area of our interest these days, because JAK inhibitors is very effective because it has multiple connected pathway, and it has shown very promising result for the management of rheumatoid, not just rheumatoid, in all sorts of autoimmune disease. Moving to the second model of autoimmune disease, the systemic lupus. This is one of the prototype of autoimmune disease which can practically affect any organ, and it does actually affect any organ. Like it can affect your brain, it can cause your ulcer in your mouth, it can affect your muscle, heart, blood, fingers, joints, abdomen, kidney, lungs, so no organ escaped. When looking into the pathogenesis of lupus, there is some apoptosis, reduced reduction of the apoptotic material clearance, so they get stored and they get expressed. So antibody synthesis, immune complex with nuclear material, complement activations. So PDC activation, interferon synthesis, B lymphocyte activation, T lymphocyte activation, again cytokine and co-stimulatory cytokines, and intercellular signaling changes. So these are the interconnected pathway of lupus that can practically affect any organ. But good part, we have at every phase, we have some kind of in our clinical practice that we can inhibit these pathways for the targeting this kind of autoimmune disease. So these are the various area where we can target. So and most of these are clinically available for the clinical practice. These are the clinical trials based on these. These molecules are already available in most of these are available. 2 or 3 of these are not still available in India, but very soon it will be available in India. The third one is a systemic sclerosis or scleroderma, where there is a more of fibrosis in the skin and muscles. So that can lead to again it at every step. So whenever there is a fibrosis, there is tissue fibroblast activation. So even VEGF, PDGF, we have options to treat all these, and we have molecules to treat this. But still we don't have the cure for any of the disease. So still our patient asks, is there any cure? We want cure, because whenever you try to suppress with one pathway, like that madam was telling, the adeno is getting converted into small cell, so there is some phenomena called immune escape. So wherever you are trying to suppress one pathway with some kind of agents which suppress a specific pathway, body tries to escape the thing, and they try to develop other cytokines and chemokines that leads to activation in another pathway and relapse of the disease. So this is something new, and possibly one of the largest hope as of now, called CAR T-cell therapy. It is very much into malignancy and cancer biology, and mostly for the leukemias and lymphomas, but it's very much coming for the autoimmune disease. So CAR T-cell are genetically modified chimeric T antigen receptors, T-cell engineered. So why CD19? As of now, CD19 is only available. So we know B cell, so there is a production of antibody. Most of the time we are having some kind of antibody production. B, we can recognize the antibody, or we are not able to recognize the antibody. There are some kind of antibody production that is causing harm most of the time. But when we are using the molecules like rituximab, which target the B cells, peripheral B cells, there are lots of B cells which are residing in the tissue, and that causes the relapse of the disease whenever there is a withdrawal of the drug or withdrawal of the effect of the drug. This is something interesting. The CAR T cell can actually inhibit your tissue-related B cells, which produce the autoantibodies and subsequent autoimmunity. So why B cell? Because autoimmune disease management has the B cell target therapies such as rituximab, they has been there for several years, but resetting the aberrant autoimmunity through deep depletion of the B cell is a potential strategy, because whenever we are using the other therapies, they are not causing the deep depletion of the B cells, so there are relapse of the disease. As of now, there are good number of trials are there, and another important, the CD19, if we go through the development of the B cell, CD19 is expressed in most of the phases, so that helps us in a more effective way suppressing the autoreactive B cell suppression. So there are large-scale clinical trials are going on for the autoimmune disease, and the results are very much promising. Patients are being drug-free remission for the years as of now, 3 years, 4 years, but it's only 3-4 years and with minimal side effects. So this possibly is the future for the autoimmune therapeutic targets and therapeutic armamentarium. So we are very much looking into that, but we don't know as of now if we suppress again this CAR T cell with the tissue depletion, what else is going to be the consequence on long-term suppression of this kind of tissue-related B cells. I'm done, sir, almost. So for autoimmune disease, the most important part, we need to treat the disease as early as possible, because if you don't use your therapeutic armamentariums early phase of the disease, there will be lots of the damage. You cannot revert any kind of damage in autoimmunity. So best therapeutic way, we don't know as of now. CAR T is showing very much promising in a therapeutic armamentarium of all sorts of autoimmune disease. So we don't, as of now, we don't have license to use for the clinical practices as on only for research purpose. These are going on, but we hope very soon it will be used. But most inhibitory factor will be the cost as of now, and this is being the ray of hope. So we need to, all autoimmune diseases, we need to catch them young. So there is a time when we started studying rheumatology 10-15 years back, then every rheumatoid has a deformed hand. Believe me, today, no rheumatoid, if he or she takes treatment regularly, there will be absolutely no deformity. For any other disease, we can absolutely prevent all kind of damage for autoimmune rheumatic disease, whatever therapeutics are available, though they are not curative, but they are very effective for controlling the disease. With this, I like to thank you. This is my alma mater, and for immunology, those who are reading and those who will be into research in immunology and rheumatology, there is no single pathway. You will have to be always observant, because there is a time when only a painkiller was a treatment for all this autoimmune disease. Now we have for every molecular level, and every day there is a new molecular pathway that is emerging. So you'll always have to be very observant for new pathways. This is the front of my school building. I don't know whether anywhere else this kind of Saraswati Murti is there. So if you notice very precisely, the toe is up, the lotus is for knowledge. So knowledge, you cannot never complete your knowledge. So always keep your thirst for knowledge, so that we can discover some more new thing and we can come up with some more curative agents. With this, thank you.
Thank you, Dr. Chattopadhyay. Very quick question, please. You can ask.
Good afternoon, sir. I have one question. Can one antigen function both as an immunogen and a tolerogen?
Yes, depending on where it is.
Okay.
Like for a very spermatozoa, when it is in the testis, it is not it's a tolerogen, it's not immunogen. But if it is present in somewhere in the body, then it is will act as an immunogen. So the protein which has to be there, because the local millili has been set in that way. So same thing can act as in both tolerogen and immunogen.
Okay, sir. Thank you, sir.
Any question?
Hello. Sir, I am Shabadadut from second year Zoology Department, Arking here. So I would like to ask you that the major problem of treating an autoimmune disorder like rheumatoid arthritis is that as our own immune cells are attacking our immune system, so when we are going to treat them, so the basic problem is become that due to the use of the drugs in a regular basis, our immune system is becoming weak day by day, and that's why the chances of being susceptible more to other diseases is being increased. So what is the another or alternative pathway for that?
So like as of now, whatever agents are available for the treatment, so from a clinician perspective, if I tell you, whenever we use like say a drug like methotrexate, most of our drugs has been borrowed from the oncology. So whenever we use methotrexate, say for 10 rheumatoid patients, 10 rheumatoid patients do not respond equally, and 10 patients do not have similar kind of side effects. So more precisely we know what exactly causing, because the first generation medicine whatever was there, they are more of a blanket therapy. So they suppress in all different pathways in a like 20%, 30% kind of. So we are moving to more towards biological therapies and targeted therapies like oncology. So more precisely we can inhibit the thing. But problem is whatever you use, every body is not going to respond in a similar way. So suppose there is a TNF elevation in this disease and you treat with an anti-TNF, but you don't know how much TNF suppression will be there in that patient. That is the problem, because that is very much individualistic. For that reason, few of the patients are doing exceptionally good. Some of them, due to oversuppression of the TNF, they develop tuberculosis. So that is the major challenge, because we don't have individualized monitoring of cytochemical and genetics. So the only way, if in future we can have a genetic mapping of the patients, which can tell us which cytokines is how much elevated, and that's why every drug do not respond to every patient. If we can have an individualized therapy for each and every patient by getting some of the genetic and chemical signature, that will be the only way out. Otherwise, till then, we'll have to use this as a blanket, whatever available is there.
Oh, thank you. Thank you, Dr.
I have a question, sir. I'll just ask one small question. Thank you, Dr. Chatterjee, for the wonderful lecture. I have just a practical question, a very basic question. You said that these diseases must be treated young. So we have to identify that this is an autoimmune disease. So how do we identify that? For example, if we have joint pains, we'll probably go to an orthopedic rather than a rheumatologist. So at what point do we know that this is not something else and we are not going to have medicines related to orthopedic or something else?
Oh, this is more practical question. So like it's more of a rheumatological domain. Orthopedics mostly deal with surgeries. For that matter, if you're having any kind of joint pain, then you can visit a rheumatologist. That is the easiest part. But problem is autoimmune disease is not just about joint disease. You can only have an eye problem with an autoimmunity. You can have only lung problem with autoimmunity. You can have only problem with your kidneys with autoimmunity. That part is very difficult to get self-diagnosed that you are suffering from autoimmune disease. Only and only if your treating physician can tell you that this is something linked to your immune related thing, then only you need to visit for the other organs. For joint related, if it's not a fracture, then visit to rheumatologist.
Thank you, Dr. Chattopadhyay.
Hello. Thank you, Professor Dasgupta, for your chair session. And now we move to the next scientific session. And so this session will be chaired by Dr. Pratap Kumar Das. And it is my absolute honor to introduce the chairperson for this session, Dr. Pratap Kumar Das, a distinguished scientist, visionary pharmacologist, and a stalwart in the field of drug discovery and translational research from Indian biodiversity. Dr. Das retired as a senior scientist from the prestigious CSIR, Indian Institute of Chemical Biology, Kolkata, where he played a pivotal role in driving innovative research at the intersection of Indian systems of medicine, reverse pharmacology, and modern biomedical science. Over the course of his illustrious career, Dr. Das led pathbreaking efforts in identification and validation of novel anti-gastric ulcer principles derived from both the plant and microbial sources. Under his guidance, one of the most notable achievements was the reverse pharmacology-based development of a hydroalcoholic extract from the flowers of Wood Florida fruticosa, a monoherbal candidate exhibiting both gastric, anti-secretory and anti-helicobacter pylori activity, the two major causes of gastroduodenal ulcers. With over 2,000 natural products screened under his guidance, Dr. Das and his team not only refined methods through high-throughput bioassays, but also steered the effort towards clinical trial readiness, including preparation of an IND doser and comprehensive evaluation of stability, safety, and phytosanitary profiles. He has 4 USA patents. His collaborative work with synthetic chemists to explore various drugs highlights his deep commitment to taking traditional knowledge from bed to bedside. So in addition to his scientific acumen, Dr. Das is admired for his mentorship, humility, and unwavering commitment to the integration of traditional Indian medicine with modern science. A very known face in the Department of Microbiology here at Ramakrishna Mission Vidya Mandir, as a passionate teacher, we are privileged to have such a visionary researcher and leader to guide us in the next scientific session. So please join me to warmly welcome Dr. Pratap Kumar Das.
Can I now ask Dr. Uday Bandyopadhyay to kindly come into the dais and make his presentations. I would introduce Dr. Bandyopadhyay very briefly instead of taking the full-scale write-up regarding Dr. Bandyopadhyay, because we have little short of time. I request Dr. Bandyopadhyay first, before I tell about Dr. Bandyopadhyay, that you have allotted 30 minutes time. If you can please make it within 25 minutes, that would be great for all of us, because the students will be able to ask few questions to you. That part is enjoyable. Regarding Dr. Bandyopadhyay, I know many things about him. I would just briefly say that he is a hardcore researcher. He has been a hardcore researcher throughout his life. He did a lot of interesting research in various fields. He has obtained almost all the awards in Indian national sciences. I would not elaborate all those things like INSA, IASC, NASA, so all these things he got, including the last one, Jagadish Bose Award. He has about 86 high quality research papers. Perhaps you have heard hearing that Google Scholar H index value is as high as 44. So he is a excellent type of researcher. I would request Dr. Bandyopadhyay to present his complicated research in a very simple manner so that students can enjoy his way of investigating the issues, so that they can feel like to be like Dr. Bandyopadhyay. That's my request to Dr. Bandyopadhyay. Make it understandable to students. Make it simple. Please, please go ahead.
Okay, many thanks for your brief introduction. In a very difficult time for me, giving lecture just before lunch to satisfy your crying gut. And second thing is that I think I will treat my lecture as small appetizer. So you will enjoy your lunch. And if someone is very angry, the easiest way to pacify or subdue the anger, to tell something, praise something about that guy. So I will tell something about stomach, I will praise a lot. So that I think all these things to be done. So my journey, I will tell you a small story. That is that it's the story of non-steroidal anti-inflammatory drugs. The drugs we are commonly using, diclofenac, aspirin, acetyl salicylic acids, every day. But the question is that these are designed to kill pain, but ultimately it will be painful. So the story of during killing pain, how it is becoming painful, I will tell you in the beginning. And again, I like to use this painful attribute of this small molecule to treat very dangerous, notorious disease called cancer. So I will use this toxic activity of the small molecule to kill a very toxic cell, that's cancer cell. So the story is by targeting the same target. So I will tell you the story of it. So these are the small painkillers, the pain, the prompt and prominent response. You cannot kill pain to rescue yourself. It is the body expresses the feeling of discomfort or complains about the fact that something has gone wrong and needs attention to reinstate a healthy state. So you cannot kill pain. But sometimes pain is so severe, it may induce heart failure, so that you have to kill pain. So pain and pain induction is very something different. So these are the classical small molecule: indomethacin, aspirin, diclofenac, piroxicam, ketofen, ibuprofen, nimesolides. So these are the therapeutic uses. And here the problems, NSAIDs are weak organic acid, and whatever they are non-ionized and lipid soluble. So whenever you consume, it diffuses across gastric mucosal cell membrane where the pH is neutral. Going inside neutral, pH is ionized and trapped. By this it cannot come out. One ionized particle cannot come out from the cell membrane. So it is accumulated, and that is the reason that very toxic materials is finally trapped and accumulated in cell, leading to cellular manifestations to kill cell death. I will tell you the story of it. So this is the pain, the painkiller, pain, a vicious cycle activated during pain management. 30 million people consume non-steroidal anti-inflammatory drugs, and 107,000 patients are hospitalized. And that is the system. The death associated with NSAID is so severe, we are not understanding during using. But for the students, I am just giving you the categories of the non-steroidal anti-inflammatory drugs. These are salicylic acid derivatives, aryl and heteroaryls, indole and indoane, anthranilic acid, enolic acid. These are the main classical things, and by these derivatives we are making some drugs. And the principle of killing pain, it inhibits prostaglandin synthase, thereby depleting prostaglandin. And thereby it induces the feeling of comfort. But the challenge came, is a very notorious scientist, he argued. He made one knockout mice without prostaglandin synthase. But there is no spontaneous generation of gastric ulcer. So, see, he challenged that it certainly inhibits prostaglandin synthase, but it must have some extra COX activity. So one person in Japan, he was very notorious. He injected prostaglandin in NSAID treated animal. If depletion of prostaglandin is a criminal for this thing, so it will deplete, it will reinstate, it's not installing. So this is highlighting, and this was my raw material to start my research in my lab. What is that? What are the non-COX activities of non-steroidal anti-inflammatory drugs? So that was my starting point, and we need to understand this thing a model. Model is that if you make a rat and orally administer 48 or 75 milligram of diclofenac or indomethacin and keep it for 4 hours, then you open the stomach, you will find perforated bleeding in stomach. NSAID within 4 hours of consumption can cause your gastric mucosal and ulcerated form. And this is the animal model, and the cellular model we are using gastric mucosal cells in Petri plate, and the Petri plate's resident cells are treated with non-steroidal anti-inflammatory drugs to follow the consequence for dealing the cell death. So by this way, I have started the model. Very interestingly, I can tell you that diclofenac concentration dependently induces gastric ulcer. An electron microscopic view, only histology will not satisfy yourself. Go for electron microscope, you will see the architecture of subcellular particles, nucleus, mitochondria, what are the consequences? So all are disrupted here, and the mitochondrial transmembrane potential depleted, and mitochondrial oxidative stress developed, mitochondrial respiration arrested. So non-steroidal anti-inflammatory drug is targeting mitochondria and thereby limiting something that may lead to cell death. That was my raw material and assumption, hypothesis that needs to be tested to make a thesis. So by this way, I started the work again. So then what happens if it is true, then is it having any relation with human cells? So using human gastric mucosal cells incubated, not in animal, in rat. It's equivalent, the same activity. You observe that it affects mitochondria by FACS analysis, that it targets the electron transport chain and thereby forcing electron to leak out. So by this way, it is affecting the function. So this is electron transport chain. Non-steroidal anti-inflammatory drugs targets the components here, and thereby the flow of the electron and meeting to be water from oxygen for univalent successive addition of electron is leading to generation of reactive oxygen species. 1 electron reduction superoxide, 2 electron hydrogen peroxide, 3 electrons hydroxyl radical for water. So by arresting this thing, it allows the leak of electron, which is highly electrophilic to molecular oxygen, forming superoxide. So we identified by this thing. So then we are really very much fortunate in our institute. We have a flux analyzer, and we analyze the fluxes of the mitochondria, and thereby identified that, if you find the control and indomethacin, one kind of non-steroidal drugs, substantially inhibited the mitochondrial functions and respirations, thereby leading to generation of electron leakage. So now what is the consequence of electron leakage? We are also fortunate, probably in Eastern India, we have installed one microscope called super resolution microscope, where you can understand, you can see in your eye the dancing of mitochondria very nicely. If you see, it will looks like a chow mein. It's a tubular structures because it is coming from bacteria. So this is the controlled mitochondria here, just like a chow mein. So if you incubate with non-steroidal anti-inflammatory indomethacin, 6 hours, 12 hours, 24 hours, you will find the fragmentation. This is called mitochondrial fission. So non-steroidal anti-inflammatory drug induces mitochondrial fission, thereby affecting electron transport chain to streamline the flow of electrons to develop ATP. Now the question is that why, what it does. So then by dissecting this thing, it activates one seizure called dynamine related protein. NSAID activate that seizure that is cutting inside the cell. So that's why by doing again microscope, you identify this is the indomethacin and control. You will find these are the protein called fusogenic mediators, the fusogenic mediators. 2 classes of proteins are there in mitochondria. One is allowing mitochondria to fuse, called fusogenic proteins, and another class of proteins allowing mitochondria to disintegrate, called fusogenic. So we nested mapped the fusogenic and fusogenic proteins along with the DRP activation excellently, that it damages the balance and giving the fusogenic proteins are upper hand and thereby addressing the mitochondria to be disintegrated. So now the question, why DRP is becoming activated? Because I am telling in a popular way, I am going in detail the science as he has asked me. So then DRP was there, but why it is now started cutting mitochondria? So now what's happened to DRP? When DRP is phosphorylated, then it can activate and it can start dropping. So who is activating DRP? Then you identify the involvement of P38 MAP kinase pathway. And this MAP kinase P38 pathway is activating DRP, and validation of efficiency of this DRP by DRP knockdown, we did the silencing of the DRP in cell. Then NSAID were not able to activate DRP and not able to initiate the fragmentation process in mitochondria by silencing studies. So what happens? Is it possible in whole animal? So we use the whole stomach. By doing histology, again we identified that DRP is highly correlated with the mitochondria and the co-localization, and this co-localization in inside, look. So this is allowing the fragmentations, and it's electron microscopic view of that tissue, and it's clearly indicated that it allows mitochondrial fission. So what happens? So P38 DRP dependent fission accompanies mitochondrial clearance. If there is abundant distorted damaged mitochondria, what will be the fate? So then mitochondria should be cleared. How? Normal process is ubiquitination. And this ubiquitinated thing is being cleared, and the mitochondrial mitophagy and ubiquitinations are correlated during this treatment along with this process. So now the question is, who will believe it? Then we started the probing by activating DRP, silencing DRP, and blocking P38 by different small molecules. You don't have to go through. So by this we identified that the statement what we are making, these are all perfect. So now the question is that, has it any impact?
whether if you treat by silence DRP by blocking P38, so then will it restore mitochondrial transmembrane potential? It is restoring transmembrane potential. So by this way, then we identified that PKC zeta regulate P38 activation, which in turn controls DRP activation, leading to mitochondrial fragmentations. So you don't have to go to these things. So I can make a synopsis of this thing, that non-steroidal anti-inflammatory drugs or painkiller, if you consume, what will happen? It will induce mitochondrial oxidative stress and mitochondrial fragmentation. Then if it is true, then it should be validated. So we started level 1 validation and then level 2 validation. What is level 1 validation? How about preventing mitochondrial fragmentation? If fragmentation is the criminal part, I will consume NSAID, but I will arrest fragmentation. Will it be possible for therapy? Or if mitochondrial fragmentation is coupled with generation of oxidative stress, can I use mitochondrial targeted antioxidant called MitoCAN? So I don't like to give it up consumption of NSAID because of my pain and arthritis, but I will use a drug that will really mop up the generation of reactive oxygen species. So this is a level 2 validation. What we observed, the fantastic thing is that MDV1 is called mitochondrial division inhibitors. It will prevent the activation of DRP. So what will happen if we inject this small molecule MDV1 before NSAID treatment to the rat? So this is then followed, what will happen? So it should prevent gastric ulcer. It should prevent mitochondrial fragmentation. It should not allow to deplete ATP. These are the facts to be done. So then we started checking of it. So then blocking or aberrant fusion largely prevents NSAID-induced mitochondrial metabolic failure. We assessed fatty acid oxidation, mitochondrial dehydrogenase, electron transport chain complex 1 activity, respiratory control ratio, ATP content, and transmembrane potentials. All the mitochondrial parameters, the diagnostics of talking a mitochondria is perfect or not. So these are all are corrected, which was deformed. Then blocking aberrant fission prevents NSAID-induced mitochondrial oxidative stress and apoptosis also. Then what will happen? So MDV prevents NSAID-induced DRP activation and ubiquitination and pro-inflammatory gene expression is there. Now the question is that MDV prevents NSAID-induced NF-kappa activation and gastric inflammation to offer gastroprotection. You know, it is even preventing the induction. This is a gastric ulcer produced by NSAID. But if you use MDV1, there is no ulcer. It's like compound. So it is preventing. So can we design a small molecule that to bypass the activity of NSAID, that divisional inhibitors type of molecule to be coined to people suffering from painkillers associated cytotoxicity. So the hypothesis which I proposed is being validated. Now the question is, the level 2 validation is that, so if mitochondria inside is generation of reactive oxygen species by leaking of electron because of abnormal fragmentations, what will happen? Can I design a molecule? So a molecule called MitoTempo. Tempo means one kind of nitroso molecule which can mop up reactive but spin molecules. So what will happen? Mitochondrially targeted small molecule, it can go inside mitochondria only, nowhere. That is a very fantastic molecule. So we injected this molecule before NSAID treatment to rat. Then we observe the same activities, whether NSAID can produce gastric ulcer or not. So what will happen? It is really mirror image like MDV1. MitoTempo dose dependently offers gastric protection. You look it. It's really highly protected. It restores transmembrane potential and release of blood. Injury index is tremendously decreased. So by this way, MitoTempo corrects NSAID-induced mitochondrial metabolic defects. MitoTempo corrects NSAID-induced mitochondrial bioenergetic crisis and inflammation and apoptosis. Now you may ask me the question, sir, how could you be sure that all the molecule MDV1 and MitoTempo is not reducing gastric hyperacidity? So whole world is looking to have a molecule which will prevent gastric ulcer but will not block gastric hyperacidity. Say ranitidine, omeprazole, Omage, Ucid, whatever the talking, we are all the time taking. These are all blocker of acid. If you block acid secretion, what will happen? Gastric microbiome will be increased, that will make to dyspepsia, and consumption of proton pump inhibitors for a long time, that will induce hyperplasia. So what will happen? The whole world is looking for a small molecule which will prevent gastric ulcer but will not block hyperacidity. So by this way, we observe that the reduction of gastric ulcer not because of the reduction of acidity, because the pH remains very low, there is no neutrality. But what lansoprazole did is increasing the pH. So by this way, the hypothesis is that we have identified a target called mitochondria that can be used for subcellularly to prevent gastric mitochondrial fragmentation and gastric ulcers. So now the question is that NSAID triggers superoxide generation, MOS hyperactivates PKC zeta P38 DRP1 axis, fragmentation of mitochondria, blocking of mitochondrial hyperfusion protects against NSAID-induced gastropathy. So hypothesis is that. Now, but still there are open question. Why mitochondria get severely damaged? What happens to the mitochondrial defense arsenal? Mitochondria have a guardian called, so many molecules are there. So it is allowing NSAID to kill it. Why it's becoming so silent? Its own defense? Why mitochondria is being subjected? That was our next question. So this question to be addressed very nicely. What will happen? The Illumina HiSec giving us a huge opportunity for us because we know nothing about the protein modifications, gene expression, profiling after NSAID treatment. So we use control tissue stomach and NSAID exposed stomach for transcriptome sequencing details. By doing this, we observe that transcriptome sequence identified one molecule called SART3, that's called mitochondrial guardian, is being targeted by totally depleted. The first time we observed that non-COX activity of NSAID, which is SART3, it targets SART3, and by this transcriptome detailed sequencing by MAT and HubGene analysis, we came to these identifications. But this is informatics and genomics. It to be tested in lab whether I am right or not. What we did, so then we, SART3 I told you is a silent information regulator 3 and it's a mitochondrial protein, class 3 histone deacetylase. What will happen? If we allow the cell to be exposed to superoxide on reactive oxygen species, it will start acetylating mitochondrially associated so many proteins. So this acetylation will induce apoptosis. So the purpose of SART3 to deacetylate to bring them to normal state. So the acetylome of mitochondria in presence and absence of NSAID is fundamental to identify that why it's defective like this thing. So by this way we started SART3 gene expression by PCR, then mitochondrial acetylation analysis, and then mitochondrial clearance by ubiquitination, SART3 protein expression, mitochondrial DNA oxidation, and mitochondrial integrity. We all assessed in tissues exposed to NSAID, which is really suggesting that non-steroidal anti-inflammatory drug with the progress of time silence the expression of SART3. So this is associated with hyperacetylation. If this is true, NSAID exposed tissue should show hyperacetylated protein in mitochondria. So then we analyze the acetylome and identified that which of the protein are acetylated are really essential for that. So these are all fission and fusogenic proteins responsible for maintaining the dynamics of mitochondria for allowing electron to flow to generate ATP. So by this way, we identified that. So if it is true, then we are fortunate to identify a small molecule present in magnolia, that's called honokiol. This molecule activates SART3. So then we ask my student, if it is true, let the mice or rat to expose to NSAID. Then you add honokiol. If silencing of SART3 is the principle for that, so honokiol will activate, so NSAID cannot damage. It will not produce any gastric ulcer. So we administer honokiol before NSAID treatment. And then we apply NSAID after 4 hours, we expose the stomach. And then what we did, again we did transcriptome sequencing and all the metabolic profiling in mitochondria. Honokiol marvelously protected gastric ulcer. And this is associated with overexpression of SART3. So this is not fluke. So by this way, we are proposing that the next generation of therapy should be like this thing, to design small molecule which will activate SART3 and let the patient to consume non-steroidal anti-inflammatory drugs. So these are the hypothesis has been tested. So then mitochondrial fragmentation accounts to pathophysiology. I can tell you So this is the first report that NSAID induces mitochondrial fragmentation leading to apoptosis, and that is responsible for the cell for inducing injury. So the NSAID also exhibit anti-cancer activity in gastric cancer, is very, very popular. So now we tested that the same SAT3 is being targeted by NSAID to kill gastric cancer. So used gastric adenocarcinoma cell and then we expose this thing. So then what will happen? Again we did transcriptome sequencing. And then half gene analysis, the metabolic profiling. This network identified that same like whole stomach in gastric cancer cells, SAT3 is being reduced drastically. And the same way, so those SAT3 is the target. So then if it is true, then we analyzed different down-regulating genes controlling metabolic and bioenergetics functions in mitochondria. So these are all, you will understand complex 1, complex 2, complex 3, fatty acid, long chain fatty acids, short chain fatty acids, all mitochondrial parameters to be tested. All hub gene analysis is indicating that NSAID is doing like this thing. So what will happen? NSAID dose and time dependently reduces SAT3 expression in gastric cancer cell. So then if SAT3, being a biochemist, understanding if it is true, better act directly. So then we need the clone and overexpressed SAT3 protein. So in our laboratory, we received the cloned overexpressed SAT3 protein and treated with NSAID directly. Very interestingly that NSAID not only prevents the transcription of SAT3, it also inhibits the acetylase activity by binding to the NAD binding site. By doing this bioinformatics and docking analysis, we came to know that this protein is really a target even in purified form. You may ask that NSAID going inside, converted into some other molecules, that molecule is interacting, but you are talking something rubbish that NSAID is doing this thing. To nullify this, we use pure recombinant SAT3 protein exposing to NSAID and demonstrated the loss of deacetylase activity, and the mechanistic studies reveal that it binds to the NAD binding site of the SAT3 protein. So by this way, then what will happen? So if it is true, then we silence SAT3, then added the amount required to kill cell, NSAID very low level. So NSAID aggravates cytotoxicity in SAT3 silent cells. So by this you also certified that the critical role of SAT3. So SAT3 silencing aggravates the cytotoxic effect of NSAID. So the conclusion I can tell you, the first time we report that non-COX activity of non-steroidal anti-inflammatory drugs. It affects the mitochondria, induces mitochondrial fission, and we are giving in the hypothesis and the new therapeutic window to design fission inhibitors to prevent NSAID-induced gastric mucosal injury and gastrotoxicity. And then NSAID by targeting SAT3 is not inhibiting gastric hyperacidity. So the purpose to develop a small molecule which will not block hyperacidity, we need acid, otherwise we cannot digest protein. So by this way, what will happen, SAT3 stimulation, blocking mitochondrial fragmentation and all the scavenging superoxide represent potential novel therapeutic strategies against NSAID-induced gastropathy. So many thanks for your patience hearing, and I am talking not that science. So these are the persons I should acknowledge. So all my students, so they probably, I do not know, they all I studied, 20 postdocs, so they are all outside. So, it is
excellent, Dr. Bandhavadhyay, nicely presenting the big story in a very small time.
Yeah, as you told.
So now let us keep it open for questions. We have really 10 minutes time.
Look, I'll be happy. Look, I'm
face
here.
Questions. He is good to face questions.
So the question is that I am here with my money to come here, and I am spending time to talk to you. But I need something to get back so that if I get new idea from you, I can start new work in my lab. So feel free to educate me, It will be very nice because you are young brain. I did my job.
Please feel free.
Feel free, you ask me whatever the question.
Express your view.
Thank you,
You
sir,
can ask me in Bengali, no problem.
Thank you, sir, for the engaging talk. I am assistant professor at Ashutosh College. I couldn't help myself to wait till the student asked the question.
Of course.
So, a very small question. So, the treatment of the cell line, which you were using as your another model, were they under starvation, like media starvation or some kind of starvation, or normal cell culture?
Normal cell.
And still they were suffering from such big mitochondrial damages?
Absolutely. If you add NSAID,
Yes, and
without that, you
Yeah.
cannot.
Okay.
And you know, I can tell you, because you people, young people should go through, the microscope was super resolution microscope. I have make a video of mitochondria because it's paucity of time. You know, when you add NSAID, mitochondria, damaged mitochondria will ask the good mitochondria, can you help me to share your DNA?
Okay.
The movement of the mitochondrial dance, you can make trajectory in video.
So related to that point, how much time does a cell take to recover from the mitochondrial damage?
If it is exposed to NSAID, it will not take any time.
Oh, it will go for apoptosis.
Absolutely.
No way to recover.
No way. Painkiller is very dangerous if you don't have to ask to manage it. Otherwise, you know, it will kill whole. I am talking about stomach, but it also affects heart. It also affects liver. But why stomach? The symptoms appear fast because stomach cells are renewing like cancer cells. Every time the bolus of food is going, it is replenishing. So the ATP requirement of cancer, gastric mucosal cells is highest in the body. And number of mitochondria present in gastric mucosal cell is highest in the body. So that's why anything damaged in mitochondria is reflected fast. That's why stomach is being affected for gastric ulcer.
Please remember the point.
Any question? So why you are so selfish, not asking me question, you know? I am old guy, tell something, you know?
Good afternoon, sir.
Yeah.
My question is, you performed molecular docking on the proteins. Did you knew the active pocket of the protein, or did you perform blind docking on that?
We use the whole protein and then develop the structures, the existing structure. So then we docked with NSAID. And then we did composition studies with NAD. There's a co-substrate required for deacetylase activity. So the propelling activities of NSAID in presence and docking confirmed, and then we did the wet lab experiments, because I do not believe bioinformatics is blindly 100%. It will give you idea. So then we use the protein and we started binding analysis by adding NAD and the displacement, a instrument called ITC. But ITC can tell you, thermocalorimetry, addition of this thing will tell you that how much is interacting and taking like this thing. So that's why we confirmed it.
Sir, you performed molecular dynamic simulation on that. So what was your conclusion after performing the simulation of the complex?
The conclusion is that NSAID interacts with NAD binding site in SART3.
Thank you, sir.
Yeah.
Good afternoon, sir. Thank you very much for your insightful lecture. So there is a question. We know that Helicobacter pylori is a microorganism that can also cause the gastric ulcer issues. So this bacteria, to protect itself from the acidic pH, they usually escape inside the hepatic cells. So is there any therapeutic strategies to prevent this escape route, to prevent the colonization of this kind of bacteria?
Fine. First I let you know there is no link of induction of gastric ulcer by Helicobacter pylori. It has been clearly proved that it has no role. So if you kill Helicobacter pylori, still if you go for biopsy and endoscopy, you will find gastric ulcer. So Helicobacter pylori is confirmed right now one acidophilic bacteria giving rise to gastritis and later on to gastric cancer. No link. And it has a protein called urease. It cleaves urea to carbon dioxide, and this carbon dioxide is giving alkaline conditions and leading, so that where it is residing, the pH is, although low, pH 5.2 or going down 2.2 in gastric lumen, but where it is residing, because of constant release of urease, the peripheral environment is alkaline in nature. So that is a mystery, and Helicobacter pylori discovery, Warren and Marshall got Nobel Prize, not for confirming responsible bacteria for leading to gastric ulcer. They identified one acidophilic novel bacteria in the world.
Thank you, sir. Thank you, Professor.
Any more question, please? If not, I will request all of you to give a great thank you to Dr. Bandyopadhyay. Please.
Thank you, sir, for a wonderful lecture. Thank you, sir, for a wonderful lecture. And it's really an honor to see two stalwart scientists whom we have, many of us here, who are now faculties, have studied their work during our initial PhD days, studied their papers line by line to understand how they think science, and then we have moved forward. Thank you, sir, for guiding us for a long time. Thank you, sir. So we now have
Few announcements from the organization.
Yeah, sure. So we now have few announcements. So as you go out from this hall, you will see there are separate veg and non-veg counters, sharp at 4 p.m. With that, thank you so much.
What is this? I can see cancer এটা ফার্স্ট ফেজ ইভ্যালুয়েশন হচ্ছে সেকেন্ড ফেজও হবে প্লিজ স্টার্ট ইউ হ্যাভ 5 মিনিটস টু ডেমনস্ট্রেট এরকম তো নয় যে এনজাইমের কনসেন্ট্রেশন
Now, this is the cell line we have taken, and we have also performed the ASC, that is adoptive T cells. Adoptive T cells, what we are doing there, it is a very interesting technique, immunotherapy, that performs nowadays in a clinical benefits. We all know it is very much beneficial, but why never use VDSC do that? Because it is not effective for solid tumors. It is not offering, it is not helping the patients, those who are suffering from solid tumors. Why? Because the solid tumors are making the immune system resistant, and means themselves resistant to the immune responses, the immunotherapies that we are creating. How? By mutating the interferon gene. Till this much we have known that. After that, so the methodology we have got, what we have taken. Okay. So if we are taking something, we are getting an observation, obviously. That is the main thing that we need. For that, here I have just written a table format, what we are getting. The parameters we have taken, VDSC to WT. WT, what is that? Wild. Yes. So it is
I hope we had a very interactive poster session, and now we are very happy to come back again to mention the name of the Best Poster Award winners. And what I've got a feedback from all the judges from the very first round as well as the second round, that it was very difficult to prioritize the best poster, but according to our rules we had to select the best three, the three groups. It is not on the rank of merit of first, second, third. It is just the three best poster awards. And so, I mean, we are very happy to announce the names. So the first, with permission from Maharaj, I will request Maharaj to kindly, deliver the prizes to the respective students. So Maharaj, please do the honor. So the first group is from the Department of Pharmacology, Bengal School of Technology, Sugandha Hugli, Rajdeep Dotto and Shumon Dotto. If you are there, please come up. Rajdeep Dotto, Shumon Dotto. দুজন তোমার হাতে আছে, তোমার হাতে আছে। রাজদীপ দত্ত। The second group is from Ramakrishna Mission Vivekananda Centenary College, Narayan Chatterjee, Raunak and Riddhu Chakraborty. Narayan Chatterjee, Raunak Pore and Riddhu Chakraborty. If you are there. Narayan Chatterjee. And the third group is from the Department of Physiology, University of Calcutta, Shayali Bhumik, Shomik Basu and Shreya Chattopadhyay. So I would request Maharaj to speak few words, and we will conclude our program with the vote of thanks by Dr. Diptendu Sarkar, followed by Maharaj.
It's very nice that all the participants and the poster presenters, it's almost about to be 5. So throughout the day you were here and you are right now. Always we see that, you see poster presentation, obviously it was a competition. প্রতিযোগিতায় যোগদান করাটাই হচ্ছে আসল কথা, and to keep up the spirit সেটাও গুরুত্বপূর্ণ, মনে রাখতে হবে। আমি নিজেও ব্যক্তিগতভাবে এই বায়োলজিক্যাল সায়েন্সের সঙ্গে যুক্ত আছি এই কলেজে। একবার ইভ্যালুয়েশন না হলেও আমি ব্যক্তিগতভাবে গিয়ে একবার ঘুরে দেখলাম পোস্টারগুলো। আমি এসে তারপর আমাদের কয়েকজন মাস্টারমশাইকে বলছিলাম যে খুবই সুন্দর, তাই তোমাদের জন্য সবসময় বলি, দিস ইজ স্পেশালি দ্য পার্টিসিপেন্টস ফর ইউ। দেখো, বিচারক মণ্ডলীর একটা ইভ্যালুয়েশন আছে, সেটা যেমন ঠিকই আছে, কিন্তু তোমরা এই যে প্রতিযোগিতায় যোগদান করেছো, নিজেদের প্রেজেন্টেশনটা রেখেছো, এটা খুব বড় কথা। তারপর তো একটা জিনিস আছে, তাই তোমাদের প্রত্যেককে যারা এখানে প্রেজেন্টেশন করেছো এবং এসেছো তাদের সকলকে অনেক ভালোবাসা, শুভেচ্ছা জানাচ্ছি এবং এইটাই ধরে রাখবে সারা জীবন। আর we always say that Vidya Mandir Paribar has no exclusion. Once you have come here, you are included. তো আমাদের এই একাডেমিক যে আজকে সেমিনার বা প্রেজেন্টেশন হলো তার বাইরেও আমাদের বিদ্যা মন্দিরে অজস্র প্রোগ্রাম হয় এরকম সেমিনার ওয়ার্কশপের পাশাপাশি। আমাদের ওয়েবসাইটটা দেখলে বুঝতে পারবে তোমরা, এই কলেজে আমাদের পড়াশোনার পাশাপাশি ছেলে মেয়েদের মানে তোমাদের প্রত্যেকের জন্য বিভিন্ন ধরনের ট্রেনিং দেওয়া হয়। আমাদের কলেজে 2টো ক্যাম্পাস আছে। তোমরা যারা যুক্ত থাকতে চাও বিদ্যা মন্দিরের সঙ্গে অন্যান্য নানানভাবে যুক্ত থাকতে পারো। আর প্রত্যেকের জন্য তোমাদের জন্য সবসময় আমাদের স্বামীজির এই প্রতিষ্ঠান দ্বার মুক্ত থাকবে। সবসময় এসো এবং আবারও যখন এরকম একটা পরিসরে তোমাদের সঙ্গে দেখা হবার ইচ্ছা রাখছি। বিশেষ করে পার্টিসিপেন্টদের বলছি, সবাই খুব ভালো থেকো, খুব ভালো করে পড়াশোনা করো, জীবনে খুব বড় হও, এই প্রার্থনা করছি। থ্যাংক ইউ ভেরি মাচ। Now officially Dr. Diptendu Sarkar, Head Department of Microbiology, will deliver the vote of thanks. Diptendu sir.
A very good evening to all. The best and beautiful things in the world cannot be seen or even touched. They must be felt by heart. Thank you is one such prayer among them. Today, I take this opportunity to put all my gratitude in the form of words. I consider it a great privilege to propose a vote of thanks to all the dignitaries who have witnessed it as a memorable and successful event. Today, my words are not enough to express the gratitude. On behalf of our Ramakrishna Mission Vidya Mandira family and Department of Microbiology and Zoology, I would like to acknowledge to our keynote speaker, Dr. Triporna Sen, Madam, from the Department of Oncological Studies, the T.C.A. Cancer Institute, who is also serving as co-director in the lung cancer PDX program at the Eaton School of Medicine, Mount Sinai, New York, United States of America. Madam keynote graced us with her thought-provoking address on novel biology and emerging therapies for lung cancer. We are really grateful to you, Madam, for being present here today and share your valuable thought on the scientific era. I would extend my heartful delight to our first speaker, Dr. Argho Chattopadhyay, who is Academic Director at the Asian Institute of Immunology and Rheumatology, Kolkata. Sir graced us with his thought-provoking address on immune disorders from basic understanding to recent advances in translational research. We are really grateful to you, sir, for being present here and share your thought on this scientific area. I am grateful to have our second speaker of the day, Dr. Uday Bandyopadhyay, sir, on board, ex-director at the administration of the Bose Institute, Kolkata, who graced us with his thought-provoking address on unique role of non-steroidal anti-inflammatory drugs to induce both normal gastric mucosal and cancer cell death, targeting SIRT3 and disrupting mitochondrial function, implication in gastric ulcer and cancer. The way sir has explained the topic is just exemplary. I would like to thank our two-session chairperson, Professor Dr. Sudhir Chandra Dasgupta sir and Professor Dr. Pratap Kumar Das sir for guiding the session, introducing speaker, managing time, facilitating discussion, and ensuring a smooth and engaging experience for attendees. I bestow my humble thanks to the respected Principal Maharaj of our esteemed Swami Mahapragyanandaji for his firm helping hands in our endeavors. And also Vice Principal Maharaj and all other monastic members for all words of wisdom what radiate from them, that is the source of energy of all of us. We will ever remain grateful to you all, and we thank you for being with us every day. I would also like to thank all faculty members, research scholars, PG students, and UG students from various universities and colleges present here and made our event more successful and colorful. Dear participants, I am hereby congratulating you all for your notable zikr and participation in this experience. You have enlightened us by your considerable presence throughout the pinnacle of our trials. I would congratulate all fellow presenters for their humble strives, while also congratulating the winners. I would like to encourage all contestants for keeping up their aspiration. There is a negligible gap between winning and not winning. What matters the most is contribution to the contest, which I believe all of you have assessed today. I also want to thank all the faculty members of Department of Microbiology and Department of Zoology for taking efforts to make this conference possible. I extend my sincere thanks to the non-teaching staff of our college and office, especially Ranjan Babu and his team, for their enormous cooperation in the organization of this type of event. An event of this dimension cannot happen overnight. The wheels start rolling months in advance. We have been fortunate enough to be backed by a team of very motivated and dedicated students committee and volunteers. I cannot thank everyone enough for the involvement they have shown and the willingness they have expressed to take on the completions of tasks beyond their comfort zones. Last but not the least, I would like to encourage applause to my dear students of microbiology and zoology of Vidya Mandira for their interest in this program and for their zeal in preparing this setup. At last, prayer to the feet of Thakur Ma and Swamiji, I would hence like to accomplish my vote of thanks. Once again, thank you all.